Okay, welcome. What can I tell you? Here we are for the 10th meeting of the NeuroImaging Training Program summer course. You are our final class of students for this, so you should be excited. I'm excited for you. Um, and We've decided to ask everybody to prepare two truths and a lie, and I think that I made that request of everybody over the weekend, so here are my two truths and a lie. They are, though I perform hypnosis, I am not hypnotizable. I'm a college dropout, and my lab director advised me to leave science. So you can think about that as you get to know me, and we'll vote at the end, and you'll decide. So let me start off by telling you just a wee bit about the training program. The genesis of this thing happened many, many years ago, now almost 12 years ago, when we lobbied successfully, as it happens, to the National Institutes of Health that there really should be a training program in neuroimaging to sort of organize people's thinking about that. Because in fact, neuroimaging was, as the way we do this stuff, was a relatively new thing at the time. So the NIH put together an extraordinary program in which they got multiple institutes within the National Institutes of Health to coalesce to add funding to the program. And they set it up so that it includes both, I'm sorry about the green highlighting here, they set it up so that it includes both the summer program that you're participating in here and also fellowships for the students. We've been here for 10 years with it, successfully renewed it, so we've had something on the order of 50 fellowships that we've given out for the program, all in neuroimaging. And there are two other programs that were funded originally with this, a program at Pittsburgh run by Sanji Kim, and a program at Harvard run by Bruce Rosen and his group. We've enjoyed continuous funding since 2006 from the National Institutes of Health. As I mentioned, there's the graduate program. The graduate program is local to the institution, and then we receive $100,000 a year to run this program, which is a large budget, and it's a large enough budget that we were able to bring in lavish speakers and put you in nice rooms and all the other stuff from here. There have been approximately 350 students trained in this classroom as part of this. To kind of put that in perspective, the Organization for Human Brain Mapping gets about 1,800 people, 2,000 people in here. So what's the right number? 2,000, geez, 3,000 now, okay. So 10% of the people in there, maybe roughly. Although there's a huge outreach from this because we also, as you know, webcast the program. We have had an extraordinary run of extraordinary students from here. I'm very, very proud of these pictures, which simply show some of the many, many faces who we've seen in here. And, it's been a gas. Really great people. Oh, I'm sorry. These numbers are all over the map. About 350 total summer students. And we've, one of the nice things about the funding for us is that even though it's a US-sponsored program, we're allowed to bring in students from all over the world. So a partial list of countries that people brought from here: Australia, Belgium, Brazil, Canada, China, Egypt, Germany, India, Italy, France, Hungary, Japan, Mexico, Netherlands, Poland, Portugal, Spain, Switzerland, Turkey, the UK, and of course the US. So that's really great. So that was a very generous thing that the NIH did. It's very unusual for them to do that. We've also have a classroom program which has been running for 10 years. There have been about 150 people gone through that. And as I say, we've given out 50 fellowships. This year's class, we had just under 100 applicants, generating, since you know what put into those things, 1,100 pages of material. Each of the application was read by at least 3 people, evaluated by the group, ranked, and just to give you some sense, On average, we have people who have 4 first authored papers, which is pretty damn good for people at your training level here. 6 papers as co-authors and 13 abstracts. So high productivity is one of the things we actually have been looking for with this. Your group is also international. So we have roughly 20% of the people are non-US in the room, which I'm also excited about with that. The program, as you know, is webcast, and just as a housekeeping matter, the webcast will include the speakers and the slides, but not the students, other than the questions that people ask. This is probably going out live right now. I assume it is. You're also welcome if, for whatever reason, you want to watch these things on your computer, but I can't say that I recommend that, since I never get any eye contact anyway. So just to orient you to our very complicated schedule. So first of all, as you look at the schedule, we are set up with a couple of different color schemes in here. The light gray represents stuff that's going on as normal classroom teaching, which is most of the content from this. We also have a couple of public keynote speakers, and for those ones we're going to move out of this classroom into a different room because we've invited a larger community of people to be here for that. And we've tried to provide time both before and after to make sure that it's easy to get to and from. In addition, we have a series of labs. The labs are in darker gray over there. They're participatory. We have a lot of software which is available on the website for download and installation. How many people here have downloaded and installed the software for the course? The majority, but not all. Okay, all right. We're not going to do a lab today, but make sure that we get that stuff installed on your computer, in the next 24 hours. We can help you with that. Okay. Then, in addition, we have a couple of group and student-based things going on in here. This year, we have decided to do a little icebreaker in the form of truth and lies, the thing you saw there before, in a small bio. We also have opportunities for students to go ahead and give brief presentations of their work, which is a really good way to start networking going. I also want to call your attention to the little button that says feedback. That will point you to a web link which has a survey form in which you can rate us all. In practice, we are not going to be using that rating as we have in prior years to design next year's course. However, we're all extremely interested in the feedback we get, and everybody who is teaching here is professional about it, cares about it incredibly, and we'll always like the feedback. Then, I'm not sure what I had in mind over here. Yes, on Thursday night, we have a group dinner. The group dinner is always a lot of fun. I edited out the pictures of people dancing on tables, but that happens, and it's great. And we really encourage you to have fun with that, because that's part of the experience of being a scientist. Getting to know everybody. The week 2 schedule has another keynote here, and I think there's another keynote here on Wednesday that are going to be outside of the classroom. These are wonderful invited speakers, and you'll enjoy that, I'm sure. We also, on the final day, have a career panel, which is mostly just sort of everybody here, well, most of the people here, other than the people in the back row, are relatively young in their careers, and we really want to foster everybody going forward, and so that's an important part. Another fun piece of this is that the program officer who is on the inside at NIH most responsible for setting up the training program funding participates, and so it's a Q&A session that's always good. On Wednesday, the second week, we set aside time to do a photo day. If you feel a need to pretty up on Wednesday morning for your photos, by all means, go ahead. We use those as souvenirs. And then finally, this year, we are going to, as group projects, we're going to be having little debate teams. We'll be talking to you more about that later. I think that our staff are going to work out exactly how we're going to organize that, but that should be fun. So a few topics that might be fun for debates. I'm just going to leave these. You've got the downloadable slides on here rather than read through all of these. But we thought a little bit about what might be kind of fun ideas that were worth talking about. But surely there will be others, and by all means, find a sense of humor in it too. You'll have more fun if you do that. Okay, so then I want to introduce you to a couple of people who will be participating throughout. First of all, you probably all met as you came in, Nadia Conway, Naxley Cortez, and Brandon Magno. I'm sorry about the typo on that name. Andrew Cho has been somebody you've probably all communicated with one way or another, who's been handling all of our software and technical infrastructure for the course. Melina Beatrice, who for many years has been the sort of heart and soul of administering the course. Right now she is out on maternity leave. Hopefully she'll be able to show up and you'll meet her. She's a great time and an incredible resource to everybody here. And then core faculty include Agatha Lenardowicz, who will be coming soon, Jesse Risman and Martin Monti. Jesse's already here. Martin has come in and out already. Oh, Martin's standing outside the door or something, doing science on the telephone, I imagine. And Susan Buchheimer in the back row. Hi, Susan. Mark Roseborough is our videographer. Hi, Mark. Make sure that this is a shareable experience with everybody here. By all means, if you have questions relating to process and setup and so on, ask any of these staff, Nadia, Naxley, or Brandon. If you have questions relating to computers, it's going to be probably mostly email with Andrew, but I believe you got his email. Did he send emails to you? You probably have that. No? Okay, I'll circulate that for you. Agatha and Martin and Jesse and Susan and myself will all be delighted to answer questions about science. Most of us will spend most of these 2 weeks in the room as much as possible. This is an incredibly dedicated faculty who we've worked with for many years in this course and who put an enormous amount of effort into this. We have an outstanding faculty of guest speakers. These are only the people who are repeat performers for us here. We have several new faculty each year, but we bring people back who have done extraordinary jobs for this. So our repeat offenders, Susan Buchheimer, Vince Calhoun, Pamela Douglas, Dan Ennis, Jack Gallant, Steve Grant, Jim Haxby, Agatha Lenardowicz, Martin Lindquist, Leo Moore, Martin Monti, Russ Poldrak, Jesse Risman, Dave Shattuck, Tali Arconi, Van Wedeen. These are all terrific high-end speakers who all they come here and have fun too. I know they have fun. Okay, and hopefully all the students do. This was one of the student groups doing a presentation in rap one year. It was great. Okay, so just to sort of do our process here, how many people think number 1 is true? There's one. That was easy. How many people think number 2 is true? Nobody's even voting, all right? And how about number 3? All right, well, so in point of fact, I am a college dropout twice. Dropped out of 2 schools. My lab director did indeed suggest that it would be a good idea if I found a career other than science, and I am highly hypnotizable, which turned out to be an amazing fact. You know, you tend to think that you're like, somehow if you're smart or something, it's not going to happen, but it's not the way it works. Okay, so we'll move right on to science stuff. Any questions come up about just sort of process administration, anything that you've seen there? Okay, good, silent group. I'm going to talk first about signaling in neurons, and I'm doing it principally from the perspective of trying to help understand what it is about the way neurons work that makes them visible to our imaging technologies. Obviously, the emphasis on this course is on functional MRI. So some of this, maybe most of it will be review for you, but maybe the perspective will be a little different. So we're going to talk about sort of the basics of neurons, how they communicate, and what are some of the energetics involved in this, and also a little bit about how information is encoded, and hopefully I'll say a few words about MRI and EEG, which are two different views of the same activity. So here are some lovely camera-lucided drawings of a number of different neurons, the number being 5. 5 is a tiny sample, of course. The representative number, it was more like trillions. But neurons are very striking cells in the body, in part because they're incredibly large, many of them. A pyramidal cell like this one might be millimeters long, just in the portion of it that sits right inside the brain. Pyramidal cells are probably the most ubiquitous cells inside human cortex. They're large, they're easy to see, and they have enormous amounts of interesting function to them. Some cells tend to be what we call stellate. They have a sort of a spherical shape to them, whereas the pyramidal cells, as you can see, have this odd sort of very polarized shape. The cell body's down here. The so-called dendrites, which are the incoming information, is all on one side of that, whereas the stellate cells have the sort of dendrites all the way around them. They capture information from a more spherical area. Many cells have this sort of bipolar configuration to them, and they're somehow integrating kind of, well, in this case up and down, but usually left and right. These gorgeous hedgerow cells, Purkinje cells in the cerebellum, are very, very striking in that they're almost planar, and yet they have this dramatic arbor in the orthogonal plane to that. What distinguishes neurons from other cells are several morphological features. One is that they have a single axon that comes out of the cell body, and the axon is understood to be the output transmission from neurons. And they have typically many, many dendrites, finger-like things out there from which they capture information. And they are also electrically excitable. Like all cells in the body, they have a potential difference, a voltage potential difference between inside and outside. The difference is that neurons can change them. So if you were to stick an electrode inside a neuron, which can be done, you can measure a voltage difference between the inside and the outside, and the neurons are typically about 1/10 of a volt lower, tenth of a volt potential difference negatively compared to the outside of the brain. And so the first thing I want to talk about is how that potential difference takes place. Here is we can imagine the inside of the neuron on the right and the outside of the neuron on the left, and inside of it are solvated ions, that is salts. So positive charge separating in the atoms from the negative charge, and some of it freely diffusing around. If you have a barrier between these two, what you'll find is that there's essentially the same number of positive and negative charges on both sides of that barrier, because it's actually quite hard to remove charge from a chemical solution. On the other hand, you can see there's a large concentration difference here. There's kind of more salt inside than outside from here. Were I to open that barrier, what will happen is that the charges will move down their concentration gradient, right? Much as if I opened a container of perfume right over here, that not too long from now, people in the back row would smell it. Things go down their concentration gradients. It's a very, very high-powered statistical function. The improbability of having everything in one place is too high, and things move to their more probable location, which is an even distribution. And they move enough that some of them will cross onto the other side of the membrane. Now you can see the membrane was designed, let me back a slide. The membrane was designed in such a way that only some of the charges are able to move across, the small ones, in this case the positive charges. And that makes the left-hand side of this thing, the extracellular space, more positive than the inside space. And that's how you generate the potential difference between the insides. It's relatively easy to calculate the voltage difference that happens as a function of concentration. This is the so-called Nernst potential. There are many charged ions, many different kinds of charged ions, and some of them can diffuse freely between the inside and the outside of the neurons, and some cannot. But principally, the positive charges are the positive potassium charges are in very high concentration inside the neuron and very low concentration outside, and the membrane is permeable to these, which ends up driving positive charges out, makes the inside of the neuron, as they say, about 1/10 of a volt negative. Other charges are also out of equilibrium. For example, sodium is roughly 10 times more concentrated outside than inside. However, the sodium ion can't get through the membrane, and so those concentration differences are sustained. Again, you can calculate this Nernst potential thing for multiple different numbers of mobile charges when you take into account the concentration outside and inside, as well as the permeability of the membrane to those. So if the membrane becomes more permeable, obviously the charges move down their concentration gradients. The cell membrane itself is a lipid bilayer, just a little micelle of fat, and it is constructed in such a way that the polarized head so fat is made up of a polar head, something that's charged, and a non-polar tail, basically a hydrocarbon tail. And when you throw this stuff all together and you shake it, what happens is that the fat tends to form bilayers. So you shake it in water, I should say. The fat tends to form bilayers in which the polar portions of it have high affinity for water, which itself is a polar molecule. And the non-polar tails have high affinity for each other, so they make this layer. And because the inside is made of fat, water doesn't go there. And so these end up being a pretty powerful barrier to charged particles, because charged things don't go through them. The membranes are impossibly thin at 5 nanometers, and yet there's on the order of 100 millivolts separating the two, which means there's a voltage gradient of 10 million volts per meter, an extraordinarily high voltage gradient, which, as you may know, means that there's an enormous amount of force. The charges are pushing at each other for this, and it's astonishing that the neurons can sustain that, or that any cell can sustain that. So one of the most interesting features of neurons is their electrical excitability. Here is a drawing of an experiment in which this is the axon, the output fiber of a neuron, and somebody has placed into it a current source, a source of electrical charge. And you can just push current into the neuron. And if you were to push, for example, positive charge into the neuron, what happens is that the inside becomes more positive compared to the outside. If you push negative charge in, the opposite happens. The inside becomes more negative. And you can measure that voltage difference with another electrode in there. So here's some data from experiments. If you take a small amount of charge, negative charge, into the neuron, what will happen is the neuron will respond by going more negative. Okay, so this is the potential measured inside the neuron. It takes a little time to change, which I'll talk about in a minute. And then you turn the current off and it goes back to its resting potential. If you put twice as much charge in, the neuron becomes twice as negative, behaves effectively linearly in that manner. If you put a small amount of positive charge in, a similar thing happens. The neuron depolarizes slightly. And if you increase the amount of charge, something else happens, which is that the neurons at some threshold become non-linear. They produce a single peak of a fixed duration and shape, which is more or less unrelated to the shape of the charge, the time course of the charge coming in. If I put in still more charge, I get the same response out. It's stereotyped. This is so-called action potential. A lot is known about the action potential. Hodgkin and Huxley, who are two geniuses for which they got the Nobel Prize, worked this all out in some great detail back in the early 50s. What they showed is that if you depolarize the neuron enough, what happens is that all of a sudden the neuron becomes permeable to sodium. The sodium, which is in high concentration outside, comes rushing inside and makes the neuron more positive. And just about as quickly, the potassium current goes up, which now is going to push the neuron the other way, right, towards more negative. And for reasons that are not entirely well known, the sodium permeability is transient. It just stays open for a small amount of time, and then those gates close. And the combination of the incoming positive charge and the outgoing charge through potassium charge ends up producing this brief action potential. So given that that's what goes on, let's kind of think about the energetics of this thing for a minute. Every time that a neuron fires, sodium comes into the cell. And we know that, a typical cell has something like 10 to the minus 12th liters of fluid in it. You can calculate that just from more or less the size of the cell. And if you have 40 millimolar sodium in there, you can actually calculate the number of sodium ions that must be in here, which is about 10 to the minus 14th mole, which is about actually 10 to the minus 14th ions. Right? I'm sorry, 10 to the 14th ions, excuse me. Get that right. The action potential changes the neuron from about minus 75 volts to about plus 55 millivolts, which is a change of about 0.13 volts. And you can calculate how much charge had to move based on the fact that the neuron has this membrane, which acts like a capacitor. A capacitor is simply an electronic device which separates charge by having an insulator, and the membrane is an insulator in here. In order to change the voltage across a capacitor, you have to put more charge in. And so you can go ahead and calculate how much charge had to move into the neurons in order to get that action potential. And that's about 10 to the minus 12 moles. Given that, so this is 10 to the minus 12 moles per square centimeter. So given that the neurons are about 10 to the minus 5 square centimeters of surface area, you're moving about 4 times 10 to the minus 17 moles of sodium, which means that the concentration of sodium is increased by about 0.1% with each action potential, which means that if you fired an action potential 1,000 times, the neuron would be depleted. And people have actually verified these kinds of numbers experimentally, which is kind of neat. So what keeps the cell from being depleted? It's a little battery. How do you charge it? And you charge that battery with something known as the sodium-potassium pump, which basically is a machine on the surface membrane of the neurons, which burns ATP, adenosine triphosphate, and makes a mechanical motion that takes sodium ions from the inside and pulls them out, and at the same time, or at a separate part of the cycle, like a two-stroke engine in a lawnmower, it also lets potassium ions in. And this, the amount of energy involved in this is really quite extraordinary, since we're now taking charged particles and moving them through a 10 million volt per meter field, right? An enormous amount of force in order to do this, but it does this by more or less exploding ATP. I guess I'll refer to this in a little bit, but this takes an enormous amount of energy, right? And you're burning sugars to make ATP, and then you're burning the ATP, and the neurons demand a lot of energy. And they need that energy to recover their resting state potential with each action potential. There's some other interesting properties of neurons. This is a sort of an electronic diagram. How many people recognize what this device is? Just raise your hand so I can see it. A few, Okay. This is a device called an electrical resistor. It resists the flow of current. This is an electrical device called a capacitor. I talked about them before. And you can model the membrane, which is this gray area here, as a combination of resistors and capacitors. And in fact, you could build a device like this with your own two hands, making it out of resistors and capacitors, or you can measure these kinds of effects. But if you were to apply a voltage difference between inside and outside, just at a point over here on the neuron, what you'd find is that if you measure the potential difference at different distances away from that point, it falls off. And that's because of the leakiness of the membrane through these resistors. Okay? So here, just where I insert the current, I get whatever is my applied, just where I insert the voltage, whatever is my applied voltage. And as I go away, it falls off. And it falls off exponentially with a spatial constant, lambda, determined by the resistors, which is, boy, I'm missing a number on here. Lambda is on the order of microns, of tenths of a millimeter to millimeters. Okay? It's pretty small, which means if you move away a couple of millimeters from wherever you're inserting the voltage, that there's virtually no potential difference created by that. That has interesting effects, which have to do with how the action potential is created and transmitted. Another feature from this is because there is a capacitance in here, that adds a time constant to the neurons. It takes time to bring charge to a capacitor. So if you apply instantaneously a step in current over here, the voltage takes a while to settle in. And the time constants for neurons is in the order of milliseconds. Okay? Relatively long. So neurons communicate with each other by making these action potentials, and then the action potentials get transmitted down the axon and where they reach other neurons. But how they get transmitted down the axon turns out to be, it's very, very well known, but it seems to be a source of confusion among many people who are in the functional neuroimaging business. The axons are not wires. It's not that they conduct charge through the inside of the axon the way a copper wire does. In fact, what actually has to happen is different. You have an action potential forming, say, right over here. And what happens is that if you measure at a small distance away from that point, the amplitude is lower because of the spatial constants of the neurons and slightly delayed because of the time constants of the neurons. And if a neuron, if the voltage is below threshold for the sodium channels to open, it doesn't form an action potential. However, immediately adjacent to my action potential over here, it looks like position V2 is going to form an action potential because it's crossed its threshold. So the neuron reforms the action potential, and then everybody else down the line starts to see a higher voltage, and the action potential propagates through the neurons. So this process of propagation to the neurons is, number one, not instantaneous. Number two, it's not like a piece of wire. The amount of current that's flowing through the neurons this way along them is very, very tiny. Most of the current flows across them. It's relatively slow because of the time constants of the neurons. So the typical velocity is only in the order of meters per second. So the distance from my brain to my knees is on the order of a meter. And my brain has to somehow communicate to my knees in time for me to move them out of the way when somebody's going to kick me. And so something has to happen over here because this is a slow process. What happens is called myelin. Myelin is actually just more cell membrane, just like I showed you before, lipid bilayers, but wrapped around itself in these large spirals. So this is an EM, a cross-section through an axon, electron microscope cross-section through the axon. And you can see actually the polar head and the non-polar tails, polar heads, non-polar tails, and so on for the membrane wrapped around itself. And because this stuff is impermeable to charged particles, what it does is increase the effective distance between inside and outside the neuron. What that does is change the capacitance of the neuron. It actually lowers it. and it also increases the resistance to current going through it, both of which have the effect of increasing the space constant on the axons and also reducing the time constant. Here's another gorgeous picture of scanning EM through myelin. And you can see the axons and the lipid layers. If you study the neurons up close with a microscope, you'll find that these myelin layers are in little segments, each of which is produced by some little pseudopod from a glial cell wrapped around the neurons. And at the intersection between the segments, things get kind of thin over here. And you actually get to the point where the axon becomes effectively directly exposed to the extracellular media, which is to say its resistance is lower over there. So what happens in neurons in order for them to conduct electricity fast is that an action potential forms at a node where it's possible to have the membrane currents be significant. And at those nodes, you'll get a high voltage potential over here. Because you have myelin around the neurons, that voltage drops off relatively slowly with distance over here. So when an action potential forms over here, it's able to recreate an action potential between the next two myelin processes at the next node. So actually, if you were to look at the voltage over here, you get an action potential here, nothing here, an action potential here, nothing here, and so on forth. And the myelin speeds all that process up. With MRI, it has, in the past few years, become possible to visualize perhaps the myelin. And this is a technology called diffusion imaging. We're going to hear significant lectures about that starting on Wednesday from here. But you can actually sort of trace out where the principal pathways are in the brain for this. The myelin, being made of fat, is relatively light colored compared to the neurons, which are made mostly of water, and that explains the difference between white and gray matter in the brain. The white matter is really just the fat in the myelin. And the connections across the brain are very dense through the corpus callosum and so on. Many of you, I'm sure all of you basically know that. One neuron communicates with another one by forming an action potential, but then the other neuron doesn't receive it exactly as an action potential. What happens instead is that small amounts of chemical transmitter go from one neuron to the other. And this was discovered and shown in great detail in 1956 by Boyd and Martin, who did this brilliant experiment where they depleted the extracellular medium of calcium. And when you do that, the little vesicles that leave the neurons don't form as well. And so you reduce the number of these events of little vesicles carrying transmitter to a vanishingly small number. And then they looked at traces of these individual events, the spontaneously occurring events, and they noticed that, oh, here's a tiny one, here's one twice its size, here's one 3 times its size, and they just counted how many times you had a small one, how many times you had an intermediate one, and how many you had a big one. And this is a histogram of the distribution of those events, which follows very closely Poisson statistics shown in the red over here, all of which tells us that these are discrete events of essentially fixed size that can occur simultaneously or not. And so this was effectively the proof that you are moving tiny particles of neurotransmitter. This is way before electron microscopy was able to see these things. What do I want to show with this? I'm going to skip that slide because I don't remember exactly what I wanted to say with it. So if you then go ahead and study in great microstructural detail what's going on when one neuron attaches to the other, is you see these entities called synapses. And the synapse is a gap between the incoming neuron and the output neuron, or the receiving neuron for this. Those gaps are tiny, but there still is membrane on both sides of it, right? So the electrical current flowing from this neuron does not directly go into its receiving neuron. However, if you look in great detail in these gaps, what you'll see is, first of all, these little packets, little spherical vesicles which are now understood to contain the neurotransmitter. Another thing you see in great profusion at the synapses are mitochondria. Does everybody know what mitochondria are? Maybe you do. Mitochondria are the energy maintaining apparatus and conversion apparatus in the neurons. The fact that you have a huge amount of mitochondria in the synapse is evidence. It's evidence that something going on at these synapses requires lots and lots and lots of energy. And then you also have this thing called the Golgi complex, which most of you know is actually involved in the formation of vesicles. So this is a machine for creating these vesicles down there. Here's what they look like under scanning electron microscope, under plain electron microscopy. You can see the vesicles. On one neuron, they're round. On the other neuron, they're sort of plate-like on here, like sickle cell-y kind of things. The different neurons, turns out, have different types of neurotransmitter in them. And it's generally thought that these sort of ovoid vesicles are containing inhibitory neurotransmitter and the circular ones are containing excitatory neurotransmitter, at least if you're talking GABA and acetylcholine, which is sort of an interesting effect. You can see the mitochondria, and you can see how a huge volume of the synapse is made up of mitochondria. And you can also see the membrane is continuous between these two neurons. Right? So they're quite separated. So the idea is an action potential comes down, and in the process of coming down, it facilitates the release of these vesicles, which then go through a small gap and reach and diffuse through that gap and reach the receiving neuron. And as a result of their going through that gap, they open pores in the receiving neuron, which then allow charge to go through and cause this receiving neuron to become depolarized and ultimately form an action potential. It takes a little time, a surprisingly long amount of time, given how fast we have to do things, about half a millisecond just to cross the synapse. And a great deal is known about this machine as well right now. And without really going too much into it, the important part, at least in terms of these older experiments and other experiments you might do as physiologists, is that this process all requires calcium. Without calcium, the fusion of the vesicles with the membrane doesn't occur. Each of the vesicles contains something we're going to call a neurotransmitter. We call it a neurotransmitter because it transmits information from one neuron to another, but almost any chemical in the body seems to be able to do that. It's really sort of an exaggeration, but there are an enormous number of things which act as neurotransmitters. The most common ones, by far, are acetylcholine and gamma-aminobutyric acid, or GABA. Acetylcholine is thought to be always excitatory. It causes depolarization of the receiving neuron. And GABA is thought to be inhibitory. It causes hyperpolarization. It moves the neurons away from firing action potentials. But evolution takes advantage of whatever chemicals are around to do whatever it can make them do. And so you see on the list, for example, of neurotransmitters, something called vasoactive intestinal polypeptide, or VIP. And it was called that because they found it in the intestines first before they figured out that it was a signaling mechanism within the brain. And many, many peptides have this effect. Okay. Now, in addition to these vesicle-based synapses, it's been known for quite some time that in insects there are something called electrical synapses, where in fact, rather than having chemicals which transmit, which carry stuff through the membrane, there are actually pores that allow the passage of current from one neuron to the next. This is what they look like in cross section, and this is what they look like in transverse section. And you can see the pores are these little circular things which have an opening in them. Electrical synapses are pretty interesting. For many years, people didn't think they took place in mammals or vertebrates, but in fact they do. There are electrical synapses in our brains, and they must do something different in terms of how they conduct things. One of them, of course, is they don't have a time gap between them. These things are actually protein channels which go from one cell to the other cell. And they form these very, very tight junctions. one of the things that's known about this, at least in insects, is they have the capability of moving current one direction only. So they act like like a diode, and would happen in the brain, which would mean that only one neuron can signal the other neuron, and not necessarily both directions. It's not clear that always happens. It's very hard to really know these. So electrical synapses have different energy requirements, of course, than the vesicular synapses, because you're not moving all this apparatus around, you're not creating neurotransmitter and so on for this. And so energetically, they're probably pretty close to silent. Another thing that is well known about the neurons is that a single vesicle transfer from one neuron to the other is not going to depolarize a neuron enough to make the receiving neuron fire an action potential. In fact, it's very unlikely that all the vesicles in a single synapse are likely to depolarize the receiving neuron enough to make an action potential. So instead, what happens is you have multiple synapses onto the receiving cell. They all have to occur essentially at the same time. The events have to occur essentially at the same time to cause an action potential to be transmitted. Each vesicle creates a single excitatory postsynaptic potential, or inhibitory, which lasts for, on the order of a millisecond, but is tiny. You have to move the neurons from about minus 75 millivolts to about minus 55 millivolts in order to get an action potential, and these things are only about 2 millivolts, so 10% as large as you would need. So for this to work, multiple EPSPs have to take place essentially at once. And by at once, I mean both they have to impact the same patch of the receiving neuron, because it's a local in here, because these have these spatial constants, but they also have to happen pretty much at the same point in time. They last long enough that if you have multiple events occurring just slightly offset, they tend to superpose. And the formal math to go from here to here is called convolution, and we'll be talking about convolution throughout the course. The dendrites on the receiving neurons tend to receive incoming information also in patches. So one set of inputs might affect this area, another set of inputs might affect this area, and so on. This is an efficient way of the neurons to aggregate information, so that different portions of the neuron might be receiving different but related information. It allows them to go ahead and form an action potential. Finally, as regards this stuff, if you look really, really closely at the dendrites, they're not just smooth surfaces, but they have these spines coming out of them. And the spines and these little red zones in here are where the actual synapses take place in these neurons. And you can see that the spines have a little bulb on them and a thin neck. Now, from the point of view of the electrical properties of this, what that does is create a high resistance to current flow through that neck, which means that an action potential here is going to depolarize this portion of it very, very effectively. So the spines become information receiving points which efficiently control how the incoming signal is carried to the output neuron. And one of the things that's now understood is that these spines come and go incredibly rapidly. Over the course of seconds to minutes, animals will regrow different spines. And this is one of the ways in which neurons adapt and learn information. Okay. How do neurons encode information since all of these action potentials are stereotyped? Well, this is an oscilloscope recording of a neuron, and during the time shown here in blue, it's receiving input. In this particular case, this is a visual system neuron, and the light is being turned on, and each of these vertical bars represents an action potential. And so here we'll just do it in real time. So what you're obviously hearing is that the neuron fires more action potentials during the time that stimulus is on. And so the rate at which this firing occurs is thought to be one of the principal ways in which the brain carries information through action potentials. Now, it could be multiple ways. First of all, the rates are somewhat limited because these action potentials are relatively, each individual one is relatively long. The neuron really under no circumstances is going to fire more than 1,000 of these per second, and more like hundreds per second when they're really maxed out. And so if you think about it, your range must be from up to 400 events a second to down to as low as what? Well, one action potential per minute would be pretty useless for our brains, right? So there's a lower range in which these things have to be firing enough for us to be carrying information. So the dynamic range must be on the order of a few per second to a few hundred per second, and that's all, right? They just can't convey much more density of information for that. The implication is that something about the brain is able to measure differences in rate with incredibly high precision. If you think about with our eyes, we can see many orders of magnitude in different light intensity by only varying the rate of these neurons firing on the order of magnitude or two, right? It's a remarkable thing. Time in the brain is a big old mystery, big old mystery. The other thing that can happen, of course, is that in addition to the firing rate of these things being a signal mechanism, each of these axons comes from someplace else. And it acts as a sort of a labeled carrier. The thing from your ear, from your cochlea, goes to a particular part of the brain, and anything that comes from the cochlea is probably auditory information. Neurons can change how effectively they allow the transmission to take place by modifying the spines, for example. That's another way your brain can change the information content. And of course, the neurons, these action potentials have to be semi-synchronous for them to do information transfer. So another way your brain can control information is the synchronicity of these events. Here's a simple example of this idea of labeled lines in place encoding. This is a schematic of the inside of your ear in the cochlea. And in the cochlea, it's known that low frequencies vibrate one portion of the basilar membrane, and high frequencies vibrate a different portion of it. And so if you have sort of a middle range frequency over here, only the middle portion of the basilar membrane moves. And so only the neurons which are innervating that portion of the basilar membrane fire signals, and therefore you can infer the frequency of the sound you're listening to based on which of these neurons fire. I mentioned this notion of inhibition. It's also possible with some transmitters to hyperpolarize the cell, move it further away from firing, and therefore they control the extent to which that neuron's going to respond to other positive going inputs for this. And the inhibitory stuff does typically require the transfer of a neurotransmitter. However, it doesn't involve the motion of sodium. It involves mostly the motion of chloride, for which there is not an active pump. So let me try and make that clear. So the chloride concentration follows the sodium-potassium concentrations for this. So producing inhibition like this does not require the pump necessarily to run more rapidly. One of the effects is that we, to this day, don't know whether inhibitory synapses are going to show up as an energetically detectable signal in things like bold imaging. We know that excitatory things do. We don't really know about inhibition. And when you see so-called negative bold effects, it's very tempting to go ahead and say, oh, well, that's an inhibitory process. You have no evidence of that, not yet. One of the interesting things about inhibitory synapses is that the majority of them in the brain are what are called presynaptic inhibitory synapses. That is, I have an incoming synapse positive, this gold one over here, and right before the signal reaches the synapse, there's an inhibitory synapse impinging on it. And effectively, if this is inhibiting this guy from firing, it gates that transmission. This was discovered first in the spinal cord, where it's part of a very, very well-known reflex process, but it is ubiquitous throughout the brain. Okay, so given all of this, what kind of things might we be able to detect using functional imaging methods or imaging methods to detect presumably as the energetics, presumably driven by things like the sodium-potassium pump and the cycling of transmitters? We can detect direct electrical signaling if you happen to be able to get an electrode near the neurons. That's kind of tough in humans, but it is done routinely now. and research is done that way. You can also measure the so-called electroencephalogram, which is the distant voltage effects from neurons inside the brain, much attenuated by the whole idea of having a skull in the distance and also blurred by those things, but you can detect it from the outside. If you have high enough resolution, you can detect differences in shapes of these neurons, which gives you some idea about their functional differences. You can also do chemical measurements from this, either direct sampling or now using a tool like magnetic resonance spectroscopy. We're not going to talk about spectroscopy in this course, but it is a window for looking at chemical concentration. There are other factors which give evidence, like how many neurons there are per cubic millimeter, and you can detect that. You could get a reasonable job at that using spectroscopy and looking at a particular chemical which is specific to neurons called N-acetyl aspartate. And then the MR image really shows a lot of difference between fat and water, which is part of the reason we see this huge gray-white contrast in the brains in the MRI. And so that gives us evidence of where the pathways are. So just briefly, and we're going to have Nikos Logothetis, who is the perfect master of this topic, come and talk to you guys about energy and neural firing. But just to get a couple of high points from here, the presynaptic neuron requires, in order for it to work, transmitter synthesis. Every time the transmitter leaves the cell, it has to be reformed, and that's an energetically demanding thing. The process of actually forming fusion with the cell membrane also requires some energy. And then in addition, this chemical which goes out into the extracellular milieu has to be scavenged for two reasons. One is that it's toxic, and the second is that it can be reused by these neurons. And so there's an active mechanism of reuptake of transmitter in the presynaptic neuron. In the postsynaptic neuron, every time an action potential forms, the battery runs down. And so you've got to drive this sodium-potassium pump. We don't really know about the inhibitory costs of these neural firings, as I mentioned, but there's some good experiments to be done. Another feature of mammalian brains is this notion of a cortical column. And this is now a very close-up view of a nose of a baby mouse. And here is little whiskers. They have a small number of whiskers that are used for sensing their environment on the order of 40 to 50 on each side. And they occur in a regular pattern from this, just like the 5 fingers on your hands occur in a regular pattern. And if you go ahead and stain a portion of the brain here on the sort of dorsolateral surface, you'll find that that you see this sort of grid pattern to certain kinds of stains. And it's known that the centers of these grids are mapped to single whiskers on the outside of the animal's nose. And all of the neurons, if you poke an electrode in here, fire in response to motion of the associated whisker. So for example, I move whisker B, and the neurons in this region all respond to this. However, you move just a little bit to the side, and they respond to a different whisker. And so in a sort of columnar way through the cortex of the brain, you see these organized functions. So that's known throughout mammalian systems. We have cortical columns associated with different places in our visual field. We have cortical columns associated with different portions of our skin and so on for this. But it's a key organizational feature of the brain. And the cortical columns themselves are on the order of a couple of hundred microns in the human brain. When we do functional MR imaging, our resolution is relatively crude compared to the cortical columns. These days, in our lab, we're scanning usually 2 millimeter isotropic voxels, which therefore contain several hundreds of cortical columns within it. And since they all have different function, we don't really get to resolve that very, very well. However, there are very high resolution techniques that people have used which seem to demonstrate that with functional MRI, you will have separable excitation of the columns. And given that functional MRI, as you probably know, and I'll be describing in a minute, is following blood flow, that implies that each of these cortical control of blood flow. And of course, each column contains thousands of neurons. We don't see single neurons with any of these tools. Why did I want to show you this? Okay, so again, our pictures of the types of neurons. I said that the pyramidal cells were ubiquitous in cortex. One of the things that's interesting about them is that the axon will be on the inner surface of the cortex, and the dendrites will be on the outer surface of the cortex. So they're highly polar cells, and they're all oriented the same way. They're all oriented normal to the surface of the cortex. A consequence of that is that here's our sort of surface of the brain and the outside of the skin. A consequence of that is that when the neurons fire action potentials, they generate a current from the cell body to the dendrites, or actually, sorry, when they're receiving input, a current from the cell body to the dendrites. However, if you have thousands and thousands of these neurons all oriented the same way, the currents are all going the same direction. And with enough of this going on, you end up with effectively a single relatively large current going through these hundreds of neurons, which is reflected as a voltage potential difference at the head. The current, the charge separation in these currents generates a voltage difference. And that voltage difference is what can be used for the electroencephalogram. It's the summated effects of many, many neurons. The EEG is problematic in so much as if you have two sets of neurons which are on opposite sides of a cortical fold, they're going to be self-canceling at the surface, which is sort of a drag. There are some other issues with EEG. For example, the signal falls off very, very rapidly with distance, falls 1 over the square of the distance. The magnitude depends on the orientation of these electrical dipoles compared to the surface. The temporal synchrony is required. We can't detect a single neuron firing from here. The neurons have to be firing in the same sort of direction at each time. Nevertheless, you can pick up a macroscopic signal. This is 30 or so electrodes on different parts of the surface of the head. And one of the prominent things you can see in here is these large sinusoidal oscillations in here. Look, this is the heartbeat, right? So you get some sense of time scale. These oscillations are occurring at about 15 times a second, 10 to 15 times a second. Does anybody know what that is? What's that called? Really? Hertz. Oh, hertz. Yeah, times per second. Okay, I'm sorry. What are these oscillations called? Alpha, yeah, alpha. Alpha is this crazy prominent thing that in brains of all mammals, you stick an electrode on their head and you start seeing these spontaneous oscillations of currently unknown function. However, just to sort of bring this full circle with this, you can measure it at the same time you do MRI. So here are some spontaneous fluctuations in alpha power in an individual. You just leave them alone and the alpha power goes up and down. You can model what the expected blood flow response is, and I'll talk about that in a minute, which might look like this red curve. And then you can find portions of the brain which mimic the power in the alpha band. And you can see now that, in fact, there are regions of the brain which very specifically increase signal intensity when alpha goes up or decrease signal intensity when alpha goes up. I show you this in part because I find it fascinating, but in part because it's also sort of linking this idea that the local electrical properties of the neurons can be proven to be associated with the bold response. I'm going to skip this just because I'm taking too long. Okay, so final slide in this section. There are many, many features of neurophysiology which become visible using magnetic resonance. And those include the energetic demands, and the energetic demands from these various components that I've talked about become visible through blood flow effects using technologies such as bold blood oxygenation level dependent signaling, arterial spin labeling, which is a flow effect. We can I'm sorry, these are all lined up strangely from this. There is the capability now, under certain circumstances, to actually image the sodium concentration. Sodium turns out to be one of the nuclei that MR can pick up. It requires relatively exotic hardware and technology, and it's a relatively low resolution, but people have done sodium imaging. And the sodium imaging might be a window into the energetics of the brain. I mentioned spectroscopy. This is a form of chemical analysis, which you can look at the chemical concentrations in individual voxels in the brain, and therefore measure things like glucose concentration or NAA concentration. There is a literature about the idea that you should be able to image the electrical currents in axons. I'm a skeptic about this. It doesn't mean these people are wrong, but I'm a skeptic because, as I said, the current through axons doesn't look like current through a wire, and you can't model it as current through a wire. But most of the models that we use in MR imaging of currents assume effectively that you have a constant current flow through the axons. Just not true. You can look at the diffusion of water molecules. We'll talk about that later using techniques such as diffusion tractography. And finally, you can look at, for example, the concentration of chemicals which are local to different cell types, and you get a map of the distribution of those cell types. Catch my breath. And I'm going to move on to this topic of a brief intro to bold imaging. And let's see, I'm supposed to go to 10:30, so this will be quick. Any quick questions you've got here? Okay, so in a couple of slides, I want to orient you to the basic bold experiment. And I want to tell you a little bit about the course design this year, which is a little different than in prior years. In prior years, we spent a relatively heavy amount of emphasis on MR physics. This year, we've mostly removed that, at least the basic physics portions of this. And that's based on the fact that most of you people claim to have a substantial amount of MRI experience. And so I'm not going to talk much about it in this slides. However, you'll see in the schedule that I have a sort of optional periods of MR physics. I love to talk about MR physics. Do it all day long. And I will do that for you. And you don't have to be embarrassed to be in that portion of the course. But if you feel like you know everything, you're in good shape, you can skip that. Okay. Functional brain studies on the basis of blood flow. This is a really cool slide. This is a fellow named Angelo Mosso, who in 1884 built a little platform that's a balance. It's got a little a bed on which somebody lies and a little edge on which they can float it, and a set of apparatus to make things balance at the center of the body. Okay? And then attached to the end of this table is a pen, which is writing on a little rotating disc. And if the balance goes down towards the head, the pen goes up, right? Simple thing. He did these experiments on people who had open skull wounds, okay? Which meant that the volume of the brain could change because there's space for the brain to go. And what he found, amazingly enough, was that if people did mathematical calculations in their head, the balance tipped downward on the head end. From which he inferred that blood flow increased to the brain when people were doing mental calculations, right? That was the first useful functional imaging experiment, as far as I'm concerned. A brilliant, brilliant thing. And he ends up quoted by many of his followers, one of whom, the famous William James, you can't say William James without saying the father of psychology, who says, "We must suppose a very delicate adjustment whereby the circulation follows the needs of cerebral activity. Blood very likely may rush to each region of the cortex according as it is most active. And of this we know nothing." So he is already aware that blood goes into the head. But he came up with the idea that maybe it depends on what part of the brain you're looking at, which is stuff he would have gotten from listening to people like Paul Broca, who had already shown that different parts of the brain do different things. The notion that you could study blood flow effects was very exciting, but it took another 100 years before somebody figured out the first way to do that. And that would have been Michael Phelps and John Mazziata at this institution, who showed using PET imaging, positron emission tomography, that the concentration of energy containing compounds, particularly in this case glucose, changed in various parts of the brain as you use them more. And this is a difference image between people looking at a blank room and watching cars go by on Westwood Plaza. And you see this large activity, for example, in the visual cortex from this brilliant experiment. The first MR versions of this were done by Jack Beliveau. Jack Beliveau tragically died 2 years ago. Jack Beliveau was a postdoc working at Harvard, we had built this ultra-high-speed scanner, and Jack got the idea that you should be able to watch blood flow in the brain using this high-speed scanner. And so what he did was arrange to inject a chemical into the arms of people, a gadolinium contrast agent, and when the chemical went through the brain went dark. And if you did this in darkness, in this blue area, you get this brief but relatively large signal change, 20% signal change or more, or in this case 25% signal change as the thing goes through the brain. On the other hand, if you do this in the presence of blinking lights to the eyes and you're measuring the signal in the visual cortex, what happens is you have a much larger signal change. And the difference between those two became evidence of activation in the brain. These were the first functional MRI experiments. Anyway, it was on the cover of a well-known magazine showing the regions whose signal changed. Now, I talked about Mosso's balance. Here's another balance. This is the balance owned by Linus Pauling. It presently sits inside the president's office at Caltech. And an interesting thing about this balance is that Pauling, who is a chemist, realized that when oxygen binds to hemoglobin and iron, it has the possibility of disturbing the spins of the electrons in the iron, a consequence of which is you change the magnetic properties of the iron. And so what he did was put blood in one of these balances, balance it all out nicely, and put a magnet beneath it. And lo and behold, the blood, when it was oxygenated, got pulled towards the magnet, whereas the deoxygenated blood did not. Phenomenal experiment. Phenomenal experiment done with, household apparatus. Pauling also got a Nobel Prize for this and a great deal of other chemical work. So like Mosso, it's understood, and if you think about the physics of it, it's well understood that if you have a magnetizable object inside the head, it will cause a signal change in the MRI, particularly if the magnetizable object is inside the blood vessels, it causes the signal to decrease. That's the basis of the Bold effect. And the changes in magnetic field with distance are very, very large when you're near a capillary, and it gives relatively large magnetic field disturbances and therefore large signal disturbances. The effect is to shorten the duration for which the signal is present in the MRI, the so-called T2 decay. And this is work done by Keith Tholborn in 1982, and this was another absolutely brilliant experiment. He realized from the Pauling studies that it might be possible to see this stuff using MRI. And in this particular case, magnetic resonance, this was kind of pre-imaging days. And he measured the rate at which the signal decays as a function of blood oxygen concentration. And you get a perfect straight line. Beautiful, beautiful experiment showing a direct effect of blood oxygen on the magnetizability of blood. On that basis, two groups, Seiji Ogawa's group at Bell Labs and Ken Kwong's group at Harvard, got the idea that you should be able to use MRI to look now at the signal changes associated with blood oxygen. And these are Ogawa's work from this, showing the blood vessels in a deoxygenated condition showing up dark, and in a hyperoxygenated condition showing up relatively bright. And the difference between those two is showing where the blood is. Where the deoxygenated blood is. These are Ken Kwong's original human data from these experiments. This is the back of somebody's head using a little receiver coil on the back of the head and in the presence of blinking lights. And this is a movie of those primary data. And you can see the signal fluctuating a little bit with noise, right? If you squint, you might be able to see that this region of the ear is dark here, and then goes bright, and then goes dark again, okay? But that's easier to see if you simply remove the mean signal. And that's what's done over here. And so you can see dark and bright fluctuations in here. This is the raw data in a good, robust MRI experiment. The very first experiments worked. Phenomenal thing. When Ken first did this, I've got to say that the other people who worked in the lab, me and everybody else, didn't believe it. Artifact, cool artifact. But in fact, he was proven right. And so in a good day, a bold experiment has such high signal-to-noise ratio that you can see the effect by eye, right? And demeaning seems like a pretty easy transformation on the data, right? And you can see it even, I mean, it's like obvious from here. So the intensity variations are known to reflect differences in blood oxygen. However, the signal goes up when you're in presence of stimulation, when you have presumably more oxygen being consumed, therefore lower MR signal because you have more deoxyhemoglobin. So why does it go up? That's sort of a paradox. And the guy who principally worked this out, I should have credited him on this slide. Oh, I can't blank his name right now. That's too embarrassing. Bob Balaban, came up with this. Oh, no, I'm sorry, I apologize. Rick Buxton came up with this theory that, and it goes like this. This is a capillary shown in gross magnification. Red blood comes in, blue blood goes out, and the reason it turns blue is oxygen flows out from the neurons into the brain. Out from the blood vessels into the brain, and it simply follows its concentration gradient. The capillaries are so thin that the oxygen simply diffuses passively across the capillaries, and it delivers enough oxygen so that the brain is kept at a relatively high oxygen concentration. Okay. Now, when the brain work increases, the oxygen content in the brain goes down. Therefore, there's a higher gradient between inside and outside of the capillaries. Therefore, more oxygen flows through. And as a consequence, what happens is that the blood turns blue more quickly because most of the oxygen has flowed out in the initial segment of the capillaries. And that starts starving the brain again of oxygen because it's all gone from the capillaries. So the response in our brain is to increase the flow of blood through these capillaries. So as you increase the blood flow, more red blood cells here you can see, what happens is that the oxygen, there is still oxygen in the capillaries by the time you get to the distal end of the capillaries. Okay? However, what's happened now is that you're delivering both high oxygen concentration by the distal ends of the capillaries, but also at high flow rates, which means now the distal end of the capillaries, now when you reach the venules, are now hyperoxygenated compared to the normal state. And the result is that you end up with a increased MR signal because, in fact, you have not more deoxyhemoglobin in the brain, but less. Okay? So that's how this happens. So when the concentration gradient is maintained throughout the capillary beds, more oxygen is delivered to the brain, but also more oxygen appears in the venules. These are, again, Ken's raw data. And here you can see, just looking at the time course in a few voxels, what happens to the signal intensity. And you saw this by eye, so it's no surprise that the signal changes are huge. In this case, a few percent. You turn the stimulus on, the signal goes up. You turn it off, the signal goes down. But none of this is actually instantaneous. And if you look at this, you can see that it sort of takes a little time to rise. I'll talk about that in a moment. There are other properties you can measure. For example, the T1 relaxation rate. Again, if we talk about physics later, you'll see this. But the T1 is sensitive not to blood oxygen concentration so much as to blood flow. And you see similar visual maps when you're measuring the T1 effects as to the T2 effects. Ogawa concentrated on the T2 effects. Ken showed both the T1 and the T2 effects in the same time, but they're very prominent. This is the basis of another form of functional imaging, which is just a flow-dependent imaging. People don't do it as much. It's slower, and its resolution is not the same. The first human brain mapping experiments, where you're going to show differential function in here, were relatively simple. You shine light in the right visual hemifield or the left visual hemifield, and you activate the contralateral portions of the brain. So here, light is turned on the, let's see, this is the left hemisphere, it's turned on the right visual field, signal goes up. If you switch the sides, you get the inversion from this. You get a very crude map of visual fields in the brain. You can look at more nuanced things. I think that this was the first experiment that truly got me excited. I mean, I participated in the original ones. I shouldn't say this is the first one that got me excited as a neuroscientist, saying we're learning something new about the brain. This is Roger Tutel's work. Roger was interested in the motion-sensitive region of the brain in monkeys called MT, which in humans is called V5. And he simply showed humans moving pictures, gratings that actually move. And he saw not only the primary visual area, but also this more lateral portion of it, which is the presumptive motion-sensitive region of the brain. Well, how did he make that presumption? What he did was study the response in that region as a function of the different stimuli. So here, we're looking at moving gratings of either low contrast or very high contrast. And in the primary visual area, you can see the signal increases monotonically as the contrast increases. In this MT region of it, the signal is constant response in the brain, essentially regardless of contrast. On the other hand, if you alternate between moving and stationary stimuli, you'll see that V1 doesn't care whether these things are moving, whereas MT, or V5, responds only if they're moving. So you actually did a functional identification of that patch of cortex. Very cool experiment. So to just sort of close up this section of here, the typical traditional form of MRI analysis is to present a task and look at the observed signal change in the brain. And if the signal changes are robust enough, like in the early visual areas, you see these large signal changes associated with the task timing. In practice, however, the signal changes are usually somewhat weaker, and they have this non-instantaneous time course. So it's more efficient to model the observed signal from first principles and then search in the brain for regions of the brain that respond like your model, right? Because your task timing is not the same as the signal response in the brain. So you want to model the time course. And the way we developed to do this was to look at what would happen if you were to just make a brief pulse of light into the eyes. And you get a time course for the bold signal which lasts several seconds and itself is much longer than the light pulse. This happens with 1/10 of a second light pulse, and yet the response is sustained for 20 seconds. But if this is the response to an instantaneous flash of light, then it turns out through basic linear systems analysis, you can predict the response of the brain to light pulses of any duration or shape. So this is now, in green, the predicted model of the brain response if we take the task model and mathematically convolve it with the impulse response model. And the convolution of those two looks like this green course over here, which you can see models incredibly well what's happening in the brain. And here, just another observation of it, the stimulus on and off, the bold signal time course, which is delayed with respect to the stimulus, and the modeled response from here. So basically all the analytic tools you use these days do some sort of convolution model in order to improve their sensitivity. This is a fun observation. And I harp on this constantly, even though this is 20-year-old work. If you change the amplitude of the stimulus, you get a scaled amplitude of the response in the bold signal. However, it's not a linear scale. In this case, we had people tapping their fingers either once per second or up to 4 times per second. And what you see in yellow are the observed responses in the brain to these different finger tapping rates, and you can see that it increases as the finger tapping rate is increased. However, these are going in log units, 1, 2, 4, and the signal intensity is not going up 4 times as much when you're getting 4 times the stimulus. Nevertheless, if you model the amplitude response as the logarithm of the signal intensity, as shown here in this blue or purple or whatever it is, it's a very, very close model to the brain. Same things happens in the visual system. You have a log, normal kinds of response. All right, with that, I have a couple of minutes to answer questions about bold and functional imaging and whatever, and if there are any, here I am. What are we going to do about this? Okay, well, I'm assuming that you guys are going to start asking questions soon enough and that I just gave you the clearest presentation you ever heard, or that you didn't understand a word I was saying. And if that's the case, come up to me after. Yeah, take the mic.
Well, I guess my question's more about the different kinds of convolution models.
Mm-hmm.
I mean, we have like some basic ones we use that are kind of available for us, but there's also some others that are out there that are kind of like custom convolution models, and
Mm-hmm.
I'm just wondering how you get those. What are some trade-offs? For example, I'm thinking about physiological signals, like peripheral physiological signals, and you
Mm-hmm.
might use some alternative convolution models rather than like double gamma.
Okay, yeah, so I was hoping to bring up a particular slide, which I'm not finding right away. Where did this come from, this particular shape, okay, that we're seeing here? And the answer is Bob Savoy, and I've forgotten her name now, Kathleen, one of his students. I'm embarrassed because this is going out in public. The two of them did a simple experiment in which they flashed a brief pulse of light in somebody's eyes and observed the response. And they did this many, many, many times and averaged them. And you get a shape. And so in order to come up with this model of the hemodynamic response function, what I did was fit their observed data to a particular low-dimensional equation called a gamma variate function. Low-dimensional meaning there's only three parameters to fit it, and one of them is an amplitude. The other two are time. And it's parsimonious to fit things with the minimum number of variables for this, right? Because the fits don't become, you don't have to correct for the number of parameters in there when you're trying to measure the models. And that particular model was done on the visual cortex specifically. However, there's no a priori reason to believe that every part of the brain is going to respond the same. In fact, the visual cortex has at least from the level of the macrovasculature, a really different architecture than other parts of the brain, that, for example, the hippocampus. Nevertheless, this model has proven to be extremely accurate in most cases. Now, if we go and look back at Ken's data, I'm sorry, quickly get there. This is again as a visual cortex stimulation. Turn the stimulation on, signal goes up, turn it off, it goes down, but it goes down below baseline. And that going down below baseline is not captured in that single unipolar hemodynamic response function. So other people, and I think Gary Glover is the one who started on this, suggested that you could add 2 more parameters to this thing, 3 more parameters, excuse me, and add another gamma variate function, which is slower and goes the opposite direction. So that the response goes up, goes undershoot and recovers. And that's the so-called double gamma model for this. Formally speaking, the double gamma model should always fit better because you've got 3 more parameters to fit, okay? And that's cheating, right? Cheating. If, on the other hand, you fix those 3 parameters, essentially in ratio to the single gamma model, then you're not cheating the same way, right? It's okay. Then it's a question of which model actually fits the data better. And this is not a trivial question because the signal-to-noise ratio in bold experiments in places other than the visual cortex is not typically this high. And so the different models will fit differently based on noise, which is always going to happen. And so whether to use one model or the other is a little unclear. I have on my computer, and maybe in the break I'll quickly come up with which slide it is, showing you portions of the brain which have an undershoot prominently and portions which do not have an undershoot prominently. In fact, the visual system has the most prominent undershoot of any part of the brain that I know of, whereas motor cortex has virtually none. So again, which model to choose? It's very tough to find a guideline. You know, you would probably be well served to do some sort of an impulse model, the equivalent of a flashing light, in whatever specific brain region you're studying, and average and average and average and average until you get a good idea of the shape, and then fit that. That's easy to do in the visual system. It's relatively easy to do in sensory motor cortex. It's really pretty hard to do in mathematical reasoning cortex, right? To have a little impulse of, oh, damn, that's an integral, quick. And so coming up with a general model for this is very, very difficult. It would be kind of nice if we could do direct electrical stimulation onto different parts of the brain and see this, but I don't see that in the offing anytime soon. The Glover double gamma model, the way it's typically shown, is actually kept this low dimensionality. You fix the ratios as these time constants. On the other hand, the various software analysis tools, for example, FSL, allows you an extremely flexible fit for this. And really what you should be doing is correcting your p-values by the number of free parameters you've allowed in that fit, right? Generally speaking, you have to go ahead and say that you have more degrees of freedom and therefore less statistical significance for any given quality of fit for these data. So that's, I mean, it's a long answer to a simple question, but that's where I am with that. Mike? I think you just turned it off.
Hello.
Oh, you're good. Okay.
So I have 2 questions. The first question is about inhibition. So in one of the earlier slides, you mentioned about negative potential that does not result in action potential.
Right.
So it's just not possible, or it's not known, kind of.
I'm sorry. The inhibition is an electrical effect. It's extremely well characterized, and it drives neurons away from action potentials. That's not controversial. What is not known is what that does to the blood flow response.
The second question I have is about myelin sheaths and the speed of response. So it seems that it's directly proportional. If your myelin sheaths are shorter, your signals will move faster across the neuron?
That's interesting. Actually, I would say in general that would be inverse to that. That is the formation of the action potential at adjacent nodes happens when the voltage in one node is high enough to increase the voltage at another node sufficient to cause transmission. Okay? If you had short internodal spacing, it's an implication really that the voltage is dropping rapidly between these nodes. And each action potential happens at a different time. This one, this one, this one, and this one. And I mean, I don't know this for a fact, so I'm only sort of hand-waving and guessing over here. But I believe what you would find is that if you studied this in real nerves, that the larger internodal spacing was associated with faster neurons and thicker myelin. Yep. It's good. We passed the mic. You don't get to hear the
I was wondering if you could talk a little bit about your opinion about directly measuring physiological dynamics in the scanner, like cardiac activity or breathing rate, to kind of model those artifacts in your signal processing, or if it's not really necessary to do that?
So we will have formal talks on that, which is a relatively broad topic. However, there are many good reasons to measure these things because they do have effect on the signal. And to a certain extent, regressing them out means you have a better model. But some of these are perfect confounds, right? So in that original Masso balance experiment, he had to correct for that. Because as more blood flow comes in with faster heart rates, that also had to be adjusted for. Anything else? Yep.
So I know the negative volt effect is like kind of difficult to interpret, but what do you do when you see these negative volt results? You just go away from them, or?
My inference is that if I'm comparing a negative bold signal to baseline, that region of the brain is is seeing less firing of the neurons. Whether it actually is as a consequence of inhibition through GABAergic cells, I can't tell. I can tell there's a cool experiment to be done. I've been wanting to do this for the longest time. You can actually inject chemicals that facilitate GABAergic transmission in the brain. You would then go and stick it into the internal carotid on one side of an animal and compare it to the other side and see what happens, right? Because you get specific GABAergic effects when you do that. But so far, I haven't been able to pull that off. It's a relatively simple experiment. Another thing you might do is associate the electrical activity in the dentate gyrus of the cerebellum, which is known to be 100% inhibitory in its outputs, with neuronal activity. Cool experiments. Just put my name on the paper. Others? Okay, so let's take a short break. There's a little bit of food back there. You can stretch. The restrooms are immediately to the left of the door in the room over here. And I guess we're good. Thanks. Just so you know, typically our speakers like to finish their slides as they're walking to the podium. So the slides for Martin's talk are up now. Also, so you know, we have many, many speakers. All of them are exceptional. All of them are famous. And we have so many of them that I actually don't really spend a lot of time introducing speakers at the course, because we'll let Martin go ahead and introduce himself. But Martin is a long-term colleague for this course, one of the course organizers, and brilliant and fun.
Hi, thank you very much. It's my pleasure to welcome you to the 10th edition of the summer program at NITP. My name is Martin Monti. I'm a professor here in the psychology department, and sometimes in neurosurgery. And I was one of you a few years ago, and then I got recruited here as a professor, and it's been great ever since. And it's been great, after having had the experience of being on your side, to have organized this for, I guess, or to have been part of this organization for about the last 5 years. And it's a lot of fun every year. You guys are different every year, which makes us different every year, and exciting every single time. So, okay, 2 truths and a lie. I didn't have too much time to think this one through, but here it goes. I was the frontman of a rock band for more than 10 years. I had artwork on exhibit at the museum, and my 7-week-old slept all night yesterday night. So go easy on me. And the night before, and the night before, and the night before. So what I'd like to do for the next sort of 50 minutes to an hour is to go over what should be a particularly simple aspect of what we do, but is in fact extremely complicated. And I think has gotten more complicated in the last 5-8 years with really this massive emergence of data of task-free type experimental designs or analysis. So I want to sort of go over what I think are some of the biggest issues within pre-processing. Let me just get a couple get you situated. Also, this slide is pretty much going to summarize the time that I will spend talking for today and tomorrow. We're going to start, obviously, imagining that you've acquired some data, perhaps some structural data, and definitely some functional data, which I'm showing here with these sort of crummy, sort of pixely brains. And then, of course, after that, you're going to do all sorts of filtering to your data. And that's what I want to talk about today. And then we're going to move on, and we're going to go from there to how to analyze that actual data, perhaps how to turn it into some kind of common space so that you can relate your own findings to previous findings and sort of explain to someone else who's not in your lab where exactly your findings happen to be in the brain, and then perhaps some method of bringing together analysis, single subject analysis, sort of from a number of individuals. If all of this has gone well, of course, you end up publishing it in Science. If this goes less well, sometimes you publish in a journal in support of the null hypothesis. Underappreciated journal for sure. I do have to make one point very clear. You might not be this kind of person. You might be this kind of person. You might like better task-free analysis, which today are so sort of ubiquitous. Needless to say, particularly the pre-processing component is just as important. In fact, if you ask me, it's even more important. So what I want to cover today is exactly this aspect of the analysis. And between today and tomorrow, we're going to go over the other boxes in here. So all the fun stuff. That's what I do. Let me allow me one slide to set my language straight, and I do it more for me than for you, because I have horrible habits when I talk about these things, and I get my language all mixed together. So I will try to abide by the following convention: that when you acquired a single, when you do a full sampling of the brain once in a functional sequence, I'll refer to that as a volume, which is the proper term. A typical run is made up of a bunch, right? A series of volumes acquired at a certain repetition. And I will try very hard to refer to this as a run. Unfortunately, I will often say scan to refer to either of these two things. If I do that and I'm unclear, stop me, ask me, I'll tell you which of the two I meant. The second intuition I want to leave you with is that throughout today and tomorrow's lectures, it makes sense to think about a run in two different ways. And different parts of what I'm going to talk about fit better one of these two intuitions. One is that a run is a bunch of sort of heads, right, acquired one after the other. And sometimes it makes good sense to think about it this way. The other way that it makes good sense to think about it is to have one head and a time series associated with each voxel within that head. And again, one of these two might be more intuitive to you, but throughout what we're going to talk about today and tomorrow, I really want you to have both intuitions in mind, because one of the two will make more sense at each of the steps we discuss. So let's dive into preprocessing. So what is preprocessing? Well, in its sort of broadest term, it's a series of data transformations, filtering, data conditioning, aimed typically at reducing sources of noise. Right? And there are some reasons why we do this, and I'll get into them in a moment. Now, with respect to task-based analysis, so your standard, task control condition type analysis, the filters that you apply and the order in which you apply them are somewhat standardized. I don't think it makes it any more trivial. I don't think it shouldn't mean that you shouldn't question the order in which you're doing things, but nobody's going to get upset at you for following the vanilla type series of preprocessing steps. Now, maybe you don't want to do that, and you might have very good reasons. There might be good data to not to do things the way we currently do, but I do get a feeling that most people are peaceful about the typical order these things happen in a task-based environment. In a task-free environment, which steps to apply and in what order is a huge can of worms. The order is a big problem, and which steps to apply depend on which kind of analysis you're doing. Right? So sometimes bandpass filtering, for example, you might want it, you might not want it. Smoothing, you might want it, you might not want it, according to what kind of analysis you're doing, how you end up segmenting the brain, et cetera, et cetera. So particularly if you're doing a task-free analysis, you have to be able to think very deeply about these steps. You have to know exactly how each of these steps is affecting your data, because it will, and there's plenty evidence to show this, it will change your results if you do, for example, graph theoretic analysis. If you're one of those people, these decisions that might seem innocent will change in a disconcerting way your results. And if you've done some of those analyses, I sure have, and you have tried it with different preprocessing streams, you will have noticed that it does make a big difference. All right? And this, we're still trying to iron this part out. So that's why I think it's just all the more important that you have a deep understanding of what's happening. So where does noise come from, if that's what we're trying to diminish? Well, all sorts of sources. Really, the two main, you can think of this, you can think of noise a bit as a penalty for what you want to do. For everything you want to do, you pay a penalty, and that comes in the form of noise. For example, you want to be alive in a world that exists, and then you get thermal noise, and that's just it, right? If you don't have thermal noise, then noise in your analysis is the least of your problem, because the world doesn't exist anymore or something like that. So that you just, you can't do anything. You're alive, and the world exists. Thermal noise. System noise, on the other hand, might be the penalty that you use for using a certain technology. And this might come in all sorts of forms, including a faulty wire that creates spikes in your images and anything. It can be all sorts of things. But this is tied to the type of hardware you're using. You then want to use the hardware in a specific way that allows you to acquire rapidly over only 2 seconds. It allows you to sample a whole human brain at some resolution. Well, you're going to pay a penalty for that. And that comes in the form of slice acquisition timing, right? And its associated issues, which we're going to get into. Even worse idea, you might want to put a human inside an fMRI. Gosh, all sorts of penalties to pay for that. You get oscillatory physiological noises, right? These tend to be at very different frequencies than those we are interested in, but they get aliased into low frequencies and sort of, I'll get back to this, and tend to be very problematic and tend to be very visible often in the signal. Even worse, you put a head inside a nice and really homogeneous magnetic field, you're going to do all sorts of bad things to that magnetic field. The sinuses, the ear canals, screw completely around with the niceness of the magnetic field that the nice people at GE, Philips, and Siemens provide for us. So we go in and spoil it by putting humans in there. Probably one of the worst ones, you put a human in there, short of nailing their ears with non-magnetic nails, obviously, to the bed, they will move. And I suspect even if you nail their ears, which you shouldn't do, but even if you did, there would be all sorts of movement related to heartbeat and sort of tugging, for example, of the blood vessels that climb up the brainstem, et cetera, and that would still create motion within what you're observing. And needless to say, there are all sorts of psychological variables that might be very important and might creep into your signal, particularly maybe in the form of slow frequencies. Alertness is bound to change over an experiment, learning effects, all sorts of things like that. Right. So these are sort of a broad brushed panorama of the kind of sources of noise. Of course, there's white noise, but we all like white noise because it's easy to cope with. Just do a bunch of trials. Now, what can you do about it? Well, there are two ways of going about this. One way is before scanning. And just before scanning, you just want to try to maximize what you got. Right? The simplest advice would be choose great technology, but this might not always be within your, you can't always sort of put your hand in your pocket and say, oh, I'd love to have a, 7 Tesla machine. So not all of us can do that. So you might be stuck with whatever technology you have at your institution. However, you can do plenty, even just by choosing, for example, good sequence parameters. You might want to choose very carefully what is the echo time that you're interested in according to what part of the brain you're interested. If you choose it poorly, you might end up with a big black blob exactly in the part of your brain that you meant to study. So that's one example. You want to be very well informed on how healthy your scanner is. I happen to have been at an institution where for a little while there was a faulty cable, but nobody was checking their own data. And we used to acquire data in batches. And then it turned out for 2 weeks, everybody had spikes in their data, which rendered it useless. But because nobody bothered to look at sort of these things, we all acquired a couple weeks worth. Actually, they all acquired. I wasn't, just by coincidence, I wasn't scanning at the time. They all acquired a bunch of useless data. I would have probably had the same problem. I just wasn't at that time scanning. So this is stuff that happens before, right? But what I want to talk about today is what happens after you've acquired the data. What can you do about these sources of noise? And really, the simplest thing to do, which you should always do, is look at your data, right? Rule 1 and rule 2 are look at your data. But after that, you can do a series of pre-processing, which is what I'll focus on. I'm going to keep to what I consider the standard pre-processing, so a number of steps. You will hear throughout throughout these 2 weeks of alternative ways of doing it. ICA is probably one of the most employed alternatives, where you actually use these data-driven methods to find signals, and then you deem them or you train a classifier to label them as noise or non-noise. And you'll get more of this later, so I won't focus on it. So let me really focus on this part here, right? So sort of the principle of detect and correct after the fact. So why do we do this? Well, 2 reasons. One is we really want to increase the sensitivity of our analysis, right? And the second one is that we want to ensure the validity of the model for what we're going to do after, which is the step after the analysis. And I've put ensuring in quotes because whether we truly ensure that we are meeting the assumptions of the model doesn't quite seem to be the case, but that's why we do this. Let me give you just a quick example on the SNR. Just imagine you have the simplest experiment you could ever do. Sort of an on-off block design, maybe 8 TRs on, 8 TRs off of some kind of task, maybe looking at faces and versus rest or versus scrambled images, something like that. You take one voxel and you look at its time course, and you can look at this time course and you might be able to tell just by eye that there's a certain signal and there's a certain noise, right? What is the signal that we're interested in? Well, obviously, the signal to us is the change, right, in the average off from the average on, right? That deflection in the data I can explain because it's what I inserted in there. It's my manipulation. So what is noise then in this graph? If that's a signal, what is the noise? Everything else, very good. Any other oscillation that is in there that is not the deflection from whatever is my average baseline to my average on, from my average off to my average on, that is all junk, is noise. I don't know why it happens. Why does the line squiggle during the on periods? I got no clue. I mean, maybe I've inserted by mistake, something in there that makes it squiggle, but given this very simple design, I have no clue why it squiggles. Why does it squiggle during the baseline? I have no idea, right? So that's my signal to noise ratio in a visual sense. Right? You can look at this other time course, and again, there's a clear signal, right? The difference between sort of the on and off periods, those green lines. We still have those high frequency squiggles that I can't explain, right? But as you can see, there also is a huge low frequency big squiggle, right? So now it's pretty obvious that as compared to before, our signal to noise ratio is very different. And notice that this is actually the exact same time course as the previous slide. I just injected artificially in there that big low frequency fluctuation, right? So really, our preprocessing is to go from something like this to something like this, right? This is what preprocessing is about. It's taking things that just appear to be as noise and trying to clean them off, perhaps by explaining them or by saying, well, this is really not relevant to my task, so I'll just do without, right? For example, this big slow frequency is really irrelevant to my task. It might be meaningful. Maybe there is a change in alertness in my subject. They really get interested and they fall asleep. Maybe my task is too long. Maybe that's what this is saying. But it's irrelevant with respect to my on-off, right? The only thing that I'm trying to explain. So we're trying to do this, right? To go from here to here. The second thing is that we're gonna then take this time series, apply a certain statistical model to it, the general linear model. I'm sure you're all pretty familiar with this. And this model just has certain assumptions about one of its terms. And we're gonna sort of go into some detail into these assumptions tomorrow. So get some coffee. But this is the second reason why, at least nominally, this is the reason why we do this. It turns out it's more complicated, but it doesn't matter. So these are the two reasons. Now, let me go through what I think are sort of some of the most salient preprocessing steps. Since someone made this point a couple years ago, these are not the only steps you might want to do. There are some additional steps. Dropping a couple TRs at the beginning is a very typical thing to do. But I want to focus on those that I think are sort of the most interesting ones. And in your slide set, you will see at the very end, there are a couple additional steps. One is brain extraction. Can I ask how many people work with populations that are not your average undergrad, so that the brain is not the perfect undergrad brain? Oh gosh, nobody scans undergrads anymore? Okay, then maybe I'll speak slightly faster to try to get to the brain extraction. We've created a software that allows gives you a slightly easier time when you have non-brains that are not the perfect undergrad, the perfect 22-year-old undergrad. And it's free, you can download it. If you use FSL, it's literally just one line. You can download it for free. Anyway, we can talk about this later. All right, so off we go. Very first one, slice time correction, right? And the idea behind slice time, as you probably know, is that in order to acquire to sample the brain in full over sort of at a rapid speed, well, to sample the brain, we don't sample it in 3D, we sample it in 2D, right? We sample the brain in every volume as a number of slices, right? So we acquire one slice after the other. And of course, it takes time to do this, right? In your vanilla epi sequence, it might take 2 seconds or 3 seconds or something like that. So this means that each slice that you're acquiring is at a different time with respect to the neural events that you're trying to capture, right, with the slice. So it might go something like this, right? Let's just assume that we had interleaved slices. I'll capture the first one, then the third, 5, right, and go on. And it'll take me exactly half the TR to capture the first half, right, of the slices. And this will take exactly 1 second, assuming a 2 seconds TR. And then once I'm done with these, I will capture all the remaining ones, all the even ones or all the odd ones, depending on how many slices you had or how you did it. Now, again, the problem is that I am trying to capture, I capture over 2 seconds the full brain, but the neural events then that sort of, the occurrence of the neural events with respect to my slices is now uneven. Right? And this is very problematic. Here's why. Imagine you had take a brain, and let's focus on a small piece of real estate inside this brain, which is blown up here. And this little piece of real estate, let's just assume that the neural events in there and the consequent hemodynamic response function generated from those neural events or associated with those neural events is exactly the same for any piece of for all this piece of real estate, right? The exact same thing is happening everywhere inside that sort of orange area. And notice that that orange area spans 3 slices, slice 15, slice 16, 17. Again, let's assume we're doing an interleaved acquisition. So here's how this goes. The black line is the HRF associated with those neural events, right? But we're sampling this black line at different times for the slices, right? For example, the neural event starts and we acquire 15, 17, and then a second and a half later, we acquire 16. Right? You've already seen that 16 is at a completely different part of the hemodynamic response function than 15 and 17. Right? And the same happens later, 15, 17, 16, 15. So as you rise, right, the slice that is captured later will always appear to have a stronger signal. Right? As it falls, you will capture 15, 17, 16, 15, 17, 16, 15, 17, 16. So as the HRF falls and you're sampling it as it falls, the slice that you're capturing last seems to fall faster. So if you now plot and interpolate your sampling of this smooth HRF, what you get is pictured in D. Right? The slice acquired latest always appears to have a stronger signal and then dies off faster than those acquired early, right? Which appear to have a comparatively lower signal but last longer. And this is a complete artifact. Right? This is junk, complete artifact. This is not what your data are, right? Complete artifact. So the idea of slice timing correction is to interpolate so that we pretend that these 3 dots were actually all acquired in the middle or all acquired at the same time. That's the idea behind slice timing correction. Right? Now, do people do this? Well, in task-based analysis, most people actually, it turns out, suggest not to do it. You can, but a lot of people say, well, look, it's not that helpful, first of all. It doesn't do that much to our data, and it does require interpolation. Why should you interpolate once more your data? You're already going to interpolate them so many times. Why should you interpolate the data one more time if it doesn't do that much? Although, of course, it might depend on your experimental design. If you have a block design, who cares about slice timing correction? But if you have a fast, random, jittered, event-related design, then it might be very important. It might worsen artifacts. By interpolating, you might smear spikes across your time course, which could be very problematic if it happened. And also, its interaction with motion correction is unclear. In fact, a lot of people think that these two, motion correction and slice timing, should happen at the same time, but just nobody has cracked that nut yet, or nobody has wanted to crack it, one of the two, probably the latter. On top of this, so on top of these negative factors, there are some positive factors. Look, first of all, we do spatial smoothing across slices. And this, if you're doing, for example, an interleaved acquisition, this really sort of moderates the problem, right? It mitigates it. Of course, if you're doing a sequential, then the bottom of the brain and the top of the brain will, the first and last slice will still be acquired far away in time. But sort of if you want perhaps the transition, you're smoothing out the transition across. So it might be more important if you're doing interleaved versus non-interleaved. Sorry, spatial smoothing may exert its effect and may diminish this effect more in an interleaved versus non-interleaved scenario. On top of that, if you're doing a task, then the suggestion is always to mismatch your task and your TR. Not only, perhaps you can always have your task either start at the beginning half the time and at the middle half the time, or maybe you can just let them roll independently, which is what people suggest. As long as your task happens sort of at all points, begins at all points throughout each TR, then you are sampling, right? Your task is starting and getting sampled in all the possible ways, so to speak. So this typically is what most people do when they have jitter event-related designs, fast event-related designs. And then on top of that, you typically, in task designs, you typically do use the temporal derivative at least, and that tends to soak up some of these effects. And there is some cost to that, but sort of it ends up in there. So for all these reasons, people often say, look, you know, do it, don't do it. It's sort of not a big deal. However, if you're in the world of task-free, then everybody does it. Right? And most of these issues that I mentioned don't change, with the exception of the mismatching of TR and task. Right? I mean, it might be happening. Sorry, with the exception of the temporal derivative, which you can't use because you don't really know what is the task or what is the cognitive processes that are happening. So this is a clear division, right, between the two fields, between the two types of analysis. Sorry. Now, I should say, so Mark mentioned Angelo Mosso's balance, and you should know that Angelo Mosso, when he was describing this, was already concerned with motion. So motion, which is probably the biggest offender in the world of functional MRI in terms of artifacts, was already a problem in the original brain sort of brain flow measuring device. And it keeps being a huge problem today too. Here's one example. I borrowed this from the FSL people. So here's a subject that is having a party during the MRI. And you can tell, right? You can look. You see it right there. Now, what's the problem with motion? Well, there are two really, really big problems. The first is that the type of signal change that motion can induce, I mean, is orders of magnitude greater than anything the brain is ever going to do in the way that we see it through fMRI. Right? So sort of the delta that motion can impose is so much bigger that it will eventually dwarf your signal of interest. And I'll get to this in a second. And second, of course, you lose the correspondence. If somebody, of course, what you're acquiring is sort of a grid, right, of voxels. And if now suddenly my head has moved, the correspondence between a voxel and a anatomical part of my brain is completely gone. So imagine a subject doing only one tiny motion, somewhere in the middle, sort of they move down. God forbid the two areas you are interested in are actually one on top of the other. And so now the neurons that were in the original area have gone out of that voxel. The neurons that were in the top area have now moved in the voxel of the bottom area, and you don't get anything, right? And your experiment is exactly half the time one process and half the time the other process. On top of that, now you really don't know, you can't really point at a piece of the brain and say, oh, this was X part or this was Y part, right? Huge problem, because that is what you want to end up claiming in the end. And here's a beautiful example from Huttel and colleagues from their textbook. Just take any epi and let's focus on sort of this this section of it. In fact, inside in the blue square. Let's just say that the numbers you're seeing here are the raw values that come out of the MRI machine when you did this experiment. At some point throughout one run in one volume, and now the subject moves by one voxel. Right? There's a translation by one voxel. Everything has moved posteriorly by one voxel. I mean, one voxel, if the voxels are 3 by 3 by 3, this is a 3 millimeter movement. Okay? Now look at the numbers. One has gone from 500 to 663. That's a pretty big jump. But notice those, for example, this one here, which is this guy, went from 89 to 507. This amount of change, nothing in the brain is ever going to do this other than an artifact, right? If you hammer visual cortex with blinking light and sort of to the point that you're making somebody almost sees, you will get a few percent signal change. Right? You're never going to get something this big. So now go back to the signal to noise ratio, right? The noise is just orders of magnitude bigger than your signal, which means your noise is going to dwarf entirely what you're interested in. Right? i.e., you can't see it, t-test, non-significant, no publication, gone. Right? Bad, we don't like that. So what we do is we do motion correction. And there's one tiny thing I really want to point out because it makes a very important experimental point. here's what you would typically see, right? So you do an analysis without motion correction, and there isn't much happening. Then you go, oh my gosh, I forgot, yeah, motion correction, click. Right? And now you do it, or you do the exact same analysis, and here it is, right? So what happened, obviously? Come on, what happened? Clearly. There you go. You removed the noise, right? You modeled, you fixed it somehow, and you've improved your signal to noise, and now you get your nice result. Bingo, fantastic, publication, science, 10, you're done. Now, however, this could happen too. You have a bunch of activations, right? And then you go, oh my gosh, or maybe it's your RA. You go like, come on, you didn't press that button. It's a very important button. Press it, redo it. And then he or she comes back to you a couple days later, ideally 5 minutes later, but sometime later, and they show you this. And now you go, oh my God, I lost all my activations. What happened? Excuse me? But I lost sensitivity. I'm sorry, yes? I got rid of the false positives. So what is the difference then between the top scenario and the bottom scenario? When must the motion have occurred? During my time of interest. If the motion happens during the task, there will be all this extra activation due to the motion that will look like it was in your task, but it wasn't. That's why you get all these extra activations. That's why it makes complete sense that when you clean off the motion, you get fewer activation. It's just you're getting the good ones. You got rid of the crummy ones. Right? So both scenarios are possible, but this makes a very important point. You want to make sure when you design your experiment that you try to decouple your cognitive event of interest from any kind of motion. It might be a good idea, for example, if you ask your subjects to respond, to have them do the cognitive task and then hold their response, their actual physical response, till, who knows, a few seconds later or something of that sort. Right? Because if you get task correlated motion, then it's difficult to tell apart which part of the activations have to do with motion and which do not. And even if you use motion correction, you might still, right, remain with motion-induced activations in there, right, and be unable to tease them out. Very important. How does motion correction work? Well, it's really a particularly sort of a very simple fix. You take a volume, you call that a reference. It might be the first, the last, the middle, the average of all your volumes. Different software do it differently. And then you take every other volume, you subtract them, you get a difference image, you square that, you get a variance image. And what you want to do is you want to move the second image so that you minimize the white in that picture, or you minimize the variance, right? And you keep moving, and you can translate it, you can rotate it, and you keep doing that until you reach a minimum of the white or the variance, right? So that's sort of the idea. What happens then is that you end up with a number of matrices that specify, if I take my ith, or let's say second volume, how do I have to adjust it so that I've minimized the mismatch between that and the reference? Right? This is simply how motion correction works. It's a least squares approach for those of you who are familiar with that term. Now, the important point is that you should always look at how much displacement happened. So for example, if you're an FSL user, you're probably familiar with these plots. Other software generate their own kind of plots that sort of convey the same information, but you should always look at them, right? For example, FSL just breaks down the translations from the rotations. Yeah? It also is nice enough to give you kind of a cumulative graph that just tells you how much displacement occurred. And what you really want to do here is look at the scale. Right? Typically, sort of the scale is one of the most important aspects. Right? And of course, if you see big jumps, right, you want to get worried about it. So this sort of at least gives you some kind of quantitative, and then you can calculate the average displacement, the frame displacement, things like that. And you can discard subjects based on this. You can say, we kept only subjects that had an average displacement of X. Right? Of course, no movement is good, any movement is bad. That's just it. But sort of people seem to abide by certain rules. You know, a few millimeters, nobody gets too offended if you keep the data. Not to mean that there isn't motion in that data, but people don't get offended when you write, oh, we've kept anyone that had less than an average of 3 millimeters movement. Jump every now and then. Right? And that's telling you something very important. That motion correction does do some things, but it still leaves plenty stuff in there. There's plenty stuff that remains in model. I'll get back to this point in a moment. But. So what can you do about motion? Well, you know, for one, you can try to prevent it, which would be great. But again, I don't know if you've been into any of these. I've put a friend of mine into this one, and gosh, he hated it. I do not know, you know, how this one feels. People actually now have businesses that will create, will do a 3D image of your head and then create a perfect thing that fits just perfectly in your head. So that then if your nose itches, you can go like this to sort of scratch it against it. I don't know. My personal experience is that anything more than this tends to sort of be hurtful, but according to which experiment, what experiment you're doing and how precise and things like that, you might want to have more sophisticated methods of preventing it. Right? If you're doing very careful retinopathy, for example, you have very different demands than somebody who is doing who is considering larger segments of the brain of activation. And not that both shouldn't have the same standard, but this is more of a traditional approach. Now, once you try to prevent it, then you got to cope with it once it happens, right? So let's just imagine that that's a time course of a voxel, and this was our task, maybe a standard on-off task. Number of ways you can do this. For one, you can just create a single regressor that pinpoints one or two volumes that had big jumps. Right? If you're an FSL user, and you should use this even if you're not, there's a little tool called FSL Motion Outliers. It's one line, FSL Motion Outliers, image. Now you can specify some more stuff if you want, but that easy. And this will actually create for you a regressor for each jump, each intensity jump that it deems to be an outlier with respect to the average signal and the variance of the signal over time. Right? It's very easy. It will automatically create a matrix with 0, 0, 0, 0, 1, 0, 0, 0, 0. And it will have one column per each such detected outlier. You can do this on the basis of frame displacements, root mean square, dvars, whichever is your favorite variable. You can change the thresholds for outlier detection, all sorts of things. But if you're lazy, just type FSL outliers image, and it'll create for you a matrix with a lot of zeros, and for each column, 1, 1. And the idea behind this is that you're trying to suck all the variance of the time point into this regressor so that it doesn't leak into your task, for example. And this is definitely a better option than splicing, which you should not do. You should not cut out, excise a TR, because that messes with the timing of things, the autocorrelation within the data, et cetera, which is very important. Okay. Now, so this would be a very simple way with one or more regressors. The standard thing that most people do is this: you take the motion that happened in each of, typically if you're doing a rigid body, each of 6 directions, right? 3 translations, 3 rotations, and you actually use those as additional regressors. Right? And the hope is that any change in variance that associates with the motion in any of these along any of these axes will get caught into the motion regressors and not into your task regressor. Now, in task-based analysis, most people obviously use this. I tend to see, but this is sort of my my experience on it, I tend to see a minimum of 6 motion regressors. Now, most people, particularly if they're FSL users, for example, where there's an automated way of adding more, will use 18 expanded motion regressors. I know AFNI has its own way of producing these too, and I'm sure SPM has something that allows you to do the same. And these additional 18 tend to be the first derivative of the motion, the second derivative of the motion, the difference between first, second derivative of the motion. The idea is that when you have a motion occur, it doesn't only affect that time point. It actually affects following time points too, in a certain way. I'll get back to this in a moment. In task-based designs, I typically do not see much sort of single time point modeling, right, the way I was mentioning earlier. Now, on the other hand, if you look at task-free analysis, then most people tend to use somewhere between 6 and 24 motion regressors. Whether these additional 18 regressors truly do something to mitigate motion is unclear. There is some data to show that they don't quite do as much as we'd like. I see fairly often sort of a single time point modeling, as I mentioned a moment ago, sometimes referred to, some people do scrubbing, and I'm not going to belabor on this because you're going to hear more about this further down. Some people also use global signal regression as a means to mitigate things that happen when you then look at correlations between time series. Of course, you know that if you use global signal regression, you're essentially mean centering everything, which also means that if you then care to look at areas and say whether something was positively or negatively correlated, it's difficult to interpret those negative correlations. Now, here's a real life example. In fact, this is one of my patients, not a good one. So these regressors down here, I think it was 4 regressors are my task. In fact, these were seeds. So time series associated with some part of the brain. Expanded motion regressors, 24. And these up here were all the outliers that FSL judged were outliers. Right? So this was actually my X matrix. And of course, if you think in stats terms, you look at this and you think, well, gee, you're losing a lot of degrees of freedom, right? Which I sure am. Of course, usually we don't test statistically at this level, so people don't get as upset about losing degrees of freedom at this first level. Our statistical analysis tends to be at the group level, so the degrees of freedom at the first level are sort of less problematic. If you analyze it has a profound impact. In fact, you notice that on the left, where they didn't use the spike, they didn't look at the outliers, you really lose a lot of the key areas that people consider being part of certain so-called resting state networks, whereas you do recover them if you include the outliers. Right? So this has the potential for being pretty important. One additional way of doing this that I think is particularly cool is to actually use what is called a prospective motion correction. And the idea is very simple and great, which is, so just assume this is your volume, and the yellow box around it is your acquisition box, right? So let's say there's an acquisition box around my head. If I move, right now I'm moving within the acquisition box, but the acquisition box traditionally doesn't move with my head, right? So that's how you lose the correspondence between voxels and part anatomical locations, etc. So the idea behind prospective motion correction is that in between TRs, you take a really quick, I think less than 80 milliseconds image, and if you notice that there's a delta, there's a big change in the position of the intensities, you infer that there has been motion. And so what it does is it automatically rotates the box to try to track that motion. And now, in a sense, you're trying to follow the head, right, as it moves inside the MRI machine. You're trying to adjust the box, the acquisition box, with the head. If you want a very nice example by Ward and colleagues, the top row shows you a certain slice before and after rotation without using the prospective motion correction. Whereas the bottom slice shows you one slice and that same slice after a motion if you have used the prospective motion correction. And you'll notice that while here the anatomical locations have clearly changed inside that slice, that has not happened in the bottom row. So it's a great idea. I've used it with some success in a recent sort of study. I was very happy with it. I think this comes default in many scanners. So if you're concerned with motion, this is something you could use. Although I'm not sure that the downsides of this type of approach have been fully explored. So I'm not sure we know whether it comes at some cost or not. We tried something last year, in fact, in the NITP, and there was some lack of clarity on whether this worked well for task-free type designs. But all right, so motion correction is great, right? It's something you really have to do. However, even after all of this motion correction, plenty of movement artifacts still remain. There's residual uncorrected motion. You've seen this in the example I gave you in the 2 clips side by side. Really, there's no way of detecting rapid movements within a scan. Really, our motion correction assumes that the subject is nice enough to move exactly in those few milliseconds between the end of one volume and the beginning of the next. if somebody were to move like this and go back to reasonably the same point or a similar point during a volume, you would never know that. So plenty uncorrected motion. Now the truth is that, and I was hinting at this before, there are spin history effects. You know at the beginning of each scan why you drop the first 4 TRs? Because it takes a certain time to entrain the protons in their sort of radio frequency, down, back up. It takes time to entrain them into this rhythm. If you now move, a given proton from somewhere in your brain will have entered a different rhythm. And just imagine that the proton hasn't had time to come back to its to alienate with the field. If you now try to inject a radio frequency pulse and you try to get the proton back down, you won't be as effective in doing it, which means that the signal that is going to be returned will be lower. So now, just because motion happened in 1 TR, you will now get a different signal, potentially lower, in 1 or 2 following TRs. So spin history effects, and there's an asterisk because the prospective motion correction, on the other hand, has the potential for diminishing this issue. Right? And of course, you might imagine that if your field is not just perfect, you could imagine voxels now sort of changing the exact magnitude of the field they're in, which again could be problematic. And of course, as I mentioned earlier, task-correlated motion is sort of one big issue. And this is something you can do something about beforehand, right? Before ever getting there. So what's the moral of the story? Hey, stop people from moving. Again, nailing their ears to the bed is frowned upon, but you can make sure they're comfortable beforehand. This is where setup time is actually very valuable, right? Make sure they're comfortable, make sure you tell them motion is a problem, train them maybe. Some people, particularly if you're working with children, some people often have these mock scanners where children play video games and every time their head moves, the video game freezes and they sort of train themselves to stay still. I don't know if you can legally, by the IRB, reward someone for not moving, but you could say, your payment is sort of contingent upon how much motion you did. I don't know. But you get the gist of what I'm trying to say. And of course, the really important part is that you want to decouple motion-prone tasks from cognitive events of interest, right? If you can, try to insert a spacing between whatever is the cognitive thing you're interested in and the actual motor response, for one example. And of course, you want to model motion out. And if needed, just reject a subject or around. And if your experiment is about the vanilla, 20-something-year-old undergrad, look, there are plenty of those where you come from, right? You can always find one more. Of course, if you're working with very, very precious populations, you flew someone in from Alaska because they had a certain genetic, mutation, then sort of, it's harder to reject someone for motion. You might try something else, but you might try to sort of bend over and backwards to try to fix it. Alrighty, so this was really sort of the main offender. Let me still go over some important points, but I think some of these you're very familiar with. Spatial filtering, known as smoothing, blurring, right? You all do this. And the idea is really that you're going to replace each individual voxel with a weighted average of that voxel and its buddies around it. Yeah? If you're not familiar with the term, I just like breaking down the full width half max, which just means that's how you communicate to other people how much you're smoothing. When you tell me that your full width half max was 5 millimeters, what you're telling me is that the width of the Gaussian that you used to smooth at half of its maximum was 5 millimeters. Right? So in my mind, I can picture a Gaussian of a certain width. Right? Now, this has advantages and disadvantages. Obviously, it increases signal to noise. Right? Because you're sort of wiping up, you're trying to clean up some of the random noise that is happening. If 2 voxels are nearby, they're probably doing something not too dissimilar. And just by averaging them in some way, you might, kind of get rid of some of that random noise, which is great. And what applies here is the matched filter theorem, right? The maximum increase that you get is if you apply a filter that is exactly of the shape and size as the signal that you care to look at. Now, what is the size and shape of the signal you care to look at? Well, that's why you're doing an experiment. So it's kind of tough, right, beforehand. So people go with rule of thumb, 5 millimeters, 4 millimeters. There isn't really a good way. People have not done this in any intelligent mode, meaning nobody has really looked back at previous studies to say, oh, okay, these are the type of activations we get, so we're gonna adapt our filter to the type of activations we expect. However, this does mean that you're either under or over smoothing every time you do this. Right? In fact, some parts of the brain you might be under-smoothing, some parts of the brain you might be over-smoothing, right? Which means you're helping some parts of the brain, you're hurting some other parts of the brain. An important reason for doing this is that it allows you to use Gaussian random field theory. We're going to talk about this later. It's one of the ways in which you correct for multiple comparisons and sort of ponder over issues of significance. And of course, by slightly smoothing, it might also alleviate the problems of matching individuals that might have different anatomy or different functional neuroanatomy. Right? Now, of course, you're reducing spatial resolution. I mean, you spent all this money to get something that gives you these really tiny voxels, and now you smooth over them. That's just terrible. And if you over or under smooth, right, you're hurting what you're looking at. If you over smooth, what happens? Very obviously. Sorry, if you under smooth, what happens? Come on, if you under smooth. If you have something that's bigger than your actual size of your smoothing, what happens? Hey, exactly. You're not getting your bang for your buck, right? You could get much bigger bang by smoothing the right size. If you over smooth, what happens? Exactly. You're bringing in activation increases, but you're bringing in junk that is not signal into your signal, which means you're hurting your signal, right? Obviously. And if you want to get a visual for sort of what happens as you smooth more and more, this is a beautiful animation from the FSL people. You can see that 0, 5, 10 millimeters, 20, right, full width, half max. There is an area between 5 and 10 where you are boosting your signal, but after that, you see that you lose activations, right, as you over smooth. And that's exactly for the reason I just mentioned a moment ago. But you know, this is sort of not too controversial. The one that is particularly important, particularly for those, particularly sort of in view of task-free analysis, is temporal filtering or temporal smoothing. Now, both task-based and task-free analysis, you can think of it this way. You're interested in oscillations that have a certain, frequency. Right? If it's a task, you're interested in stuff that goes up and down and wiggles at the frequency of whatever your task is. If you're interested in, if you're doing your standard the resting state kind of analysis, typically people seem to be particularly interested in the low end, right, of the fluctuations of neural events. So in both cases, you are doing this kind of smoothing. Now, you're interested in a specific window within the frequencies that you get, but of course, there's a lot of other stuff that creeps in there. And I mentioned, a lot of these earlier. I should say, currently in fMRI, most of the noise has this 1/f, so you get a lot of noise at the lower frequencies. Equipment-related noise, now this can come at any frequency, right? It could come at any frequency, but I would say most of the times I see this, we're at the very low frequencies. Now, the physiological ones are complicated. You get cardiac respiration, maybe between 40 and 120 beats per minute, or 12 to 20 breaths per minute, which gives you certain ranges, but they always end up aliasing into the low frequency because we're sampling slower. And so we tend to end up we're sampling these events slower than they are, so we end up seeing these slow fluctuations. So even sort of higher signals end up at the lower end of your frequencies. And of course here, as our TRs get faster and faster, it might be easier and easier to use these type of filtering to get rid of, for example, physiological noise. Motion, as I showed earlier, all over the place, but we have ways for coping for that. So here's a visual for it, right? Imagine you have a task. Green is what you're interested in, right? Your task might be happening exactly every 30 seconds. I have a period of 30 seconds. And then maybe it has some harmonic higher up. But the noise, what you're interested in is whatever happens somewhere around here or somewhere around here. But the noise, of course, right? You get noise all over the place. Particularly, you get noise up here, right? So the noise that I circle here in white, that's kind of easy, right? This is almost evenly distributed over all these frequencies. So it's easy to cope with just by averaging. What is difficult to cope with is structured noise, because this, you don't get rid of it just by averaging. So we got to do something about it, and that something is temporal filtering. Just by the way, if you just look at this, you can already tell that this example makes clear that the signal-to-noise ratio, right, is terrible, certainly smaller than 1, right? The noise is dwarfing in terms of its power your signal. Here's an example. This is our this is our time series that I took, that I showed you earlier. And let's just say that we have, let's add in there, right, a linear drift, which might happen. And now your signal is this. Or maybe you might want to insert in there, you know, a sort of a U-shaped drift, right, low drift. And now your signal might look like this. Or maybe you might have in there excessive high frequencies. And now your signal looks much more wiggly than before, right? So what we want to do is we want to get rid of all these things that are not pertinent to our task and that we might explain in terms of motion, drifts, things like that. Right? So the idea is to use, for example, a high-pass filter. The idea behind a high-pass filter is pretty simple. You literally say, you take your time course, you look at the power spectrum, and you say, can I just ditch anything that is irrelevant to my task, i.e., anything that is slower than a certain number is probably unlikely to be due from my task. It's more likely to be due to all these other sources. Can I just get rid of it? That's exactly what a high-pass filter does. Right? This is the shape of a high-pass filter. So take that last box on the right. Imagine that being a knife and cut off in the middle graph anything that falls outside, right, that area. So essentially what you're doing now, you're cutting off all this crap up here. Notice again that your signal to noise ratio is terrible. Right? Now apply a high-pass filter, and now this is what your time series looks like. Right? And again, you've lost sort of this big drift. Right? And indeed, if you take the power spectrum of your new time series, there you go. Now your signal is actually the power peak. So now your signal to noise is much better in terms of the power at that frequency. If you want sort of a real-life example of a event-related design, it looks very different. This is a jittered event-related design. It's less easy to tell where my task is. Right? In a nice block design, you know exactly the frequency of your task. Right? It's a periodicity of your on-off sort of blocks. In a jittered event-related, it's much more difficult, right, to find what is that number. There is software that will help you find out what it is, but the idea is the same. So here's the time course. This is a power spectrum, apply a high-pass filter, and as you see, you're kind of getting rid of this initial, right, big spike there, which you know is unlikely to be due to your design. The low-pass filter is exactly the same thing, but the other way around. Right? You get rid of stuff that is too high to be of interest to you. For example, if you have this artificial sort of high-frequency fluctuation that I've put into the signal here, you look at the power spectrum, there's your task right here. And this stuff here is crap that I injected in there artificially, just to make this look really squiggly. Yeah? So the low-pass filter, it has a slight different shape in terms of its rollout is less steep than the high-pass filter. But the idea is the same, right? Imagine this is a knife that sort of gives a number of 1 to anything here, and then lower and lower numbers until it's really nearby 0. Essentially, you can put it here, and you're kind of cutting off, right, anything that is too high by whatever you set the low-pass filter. So now you do that, and there you go. We're back to our original, right, nice time series. And if you look at the power spectrum, yet again, right, we get this, we have the peak of our task, but we lost sort of that high frequency that was not relevant to our task. And of course, I can do this because I know that that cannot be due to my task. And so I try to explain it, say, well, that's probably noise from the equipment, so let me just cut it off. Right? Let me just excise it. Okay. I'll just skip over this thing. This is just how this gets implemented. And I'll just say that there are two different ways of doing it. One is the SPM way, which essentially is just saying we will add low frequencies inside the design matrix, and those low frequencies will get captured by these regressors. And you can see here an example of the type of regressors that get added to your X matrix. FSL has a different philosophy usually than SPM. They like doing data-driven stuff. So they actually go in and say, in the data, and say, let me see what low frequencies are in here. And by using a Gaussian weighted running line, you essentially end up seeing that this line right here, the pink line, is the low frequency, and then you remove that. Right? And to give you an example, you start from the red line, and after you've removed the low frequency, you get the green line. And you can see, for example, that the time series here got dragged a little bit down, right? And let's see, yeah, and here it got dragged a little bit up. So this is the type of adjustments that you can do. So only because a lot of people, yeah, great. I will finish with one thing about brain extraction. I've left these slides that I won't really cover in the handout. But let me point this out. If you're not doing, if you're not doing analysis on your standard, very nicely shaped brain, brain extraction can be problematic. The idea behind brain extraction is to separate what is brain tissue versus non-brain tissue, right? And this is often used for a lot of, sort of underlies a lot of analysis. Fairly typical to do this in certain software packages for functional data too, but if you're doing structural data, for example, this is very important. Now this becomes very difficult if you get brains such as those that I typically look at. And these are 3 different patients, and you can notice that the shapes are not quite your standard average undergrad. I mean, some might, but, or you might have, one particular problem that we often get is sort of big enlargement of ventricles, and so that throws off a lot of the tools. If you were just to take, for example, these 3 brains and put them into the conventional tools that you get, this is the result. In gray, you're seeing the result of the tool. In green, you're seeing a manually sort of a manual brain extraction. So the perfect brain would be the one that matches, the perfect extraction would be the one that matches perfectly the green grid. Yeah? You know, not quite what you want. I mean, this one here, for example, I took off the name because it's sort of, I didn't want to do bad advertisement. But here you have all the neck, which you don't really need, or here you might have all this excess out here, right, which you don't really need, or all this growth out here, which is clearly non-brain. However, if you sort of, with some engineering, you can end up doing a pretty good job automated. So one way is that you actually do these manually, but it's very time intensive. So if you use if you see populations that sort of are difficult to work with, there's this software which you can download for free. There's a link down here, and there's a paper that's going to tell you all about it. But you can get some pretty good brain extractions in an automated way. So if you don't have the time to actually do it manually, to correct it, there is some extraction. The reason it works so well is the following. Here's how it actually works. By the way, OptiBet is the name. And you can find it somewhere on the FSL website too. Imagine you start with this brain, and you can notice this is the head that has sort of this, all right, this big deformation here. What we do is the following. We do an initial brain extraction, an approximate one of those that is crummy. See all this stuff out here that is not brain, that is still in there. Then we register it with a linear and nonlinear to the MNI template. We register the bad brain extraction. And then we take an MNI mask, one of those that FSL gives you or that you can find anywhere online. And we bring that and we apply the inverse to it. So now we take the nice mask and we bring it back into the space of the head. And then we simply punch out, right? We do a punch out from the original head image with the inverted mask. And then you actually get a pretty good, right? You can see here that now we've lost all the stuff that was hanging out on the sides. So if you don't have the time to manually adjust it, this is a very neat little tool that you can use. If you have FSL installed on whatever cluster you use, just download that script and type one line, optibet.sh image. And that's it. And if you want to read all about it, there's a paper, and the script itself will give you all sorts of instructions. And yeah, I'm going to leave you to this. So I'll gladly take questions, or maybe I'll take lunch. Well, what am I taking? I'll take some questions if people have them. But I'm around for sort of most of this too, so if you feel shy, you can ask me later.
We've left an hour and a half break in here. We tried for lunch. There's plenty of time for that. And we're really encouraging questions.
Yep, take the mic.
Is it working? So I have going a bit out on a limb here with this question about task-relevant stochastic noise. So the idea that the internal white noise in the system, if it matches the frequency of the task, it could push below threshold processes up to perform the task. And so do you know if there's a way that that can be modeled separately from the task-based signal and also from the noise that you're discarding?
So if it's white noise, by definition, it shouldn't, right? White noise, by definition, should be fairly even across all frequencies. But I understand what you're saying. What if you do have other things? Once it's close to your task, I could imagine using things such as added regressors, for example. If you measured cardiac, if you actually measured with a pulse oximeter or sort of, the breathing, then you could use that to try to get rid of structured noise of that sort. If it matches your task, it becomes very difficult because anything you get out of there could either be your task or it could be, I don't know how you would tell apart which part of it is task and which part of it is non-task, unless you have, for example, additional regressors. Even if you used an ICA and just say, yeah, show me everything that is there, unless you've already trained, here, I could see this happening. You've previously trained a classifier using one of the available tools, which I think we're going to discuss later on, to recognize what is noise and what is not noise in your typical, in your scanners, in your scanner. And then it might label some part of that as noise, but I don't know how it would tell apart which part came from the task and which came from the noise. So once it matches, it's a problem. Actually, you know what? Even if you use the regressor as the cardiac, once it matches, you're going to end up losing most of your signal into that. So the answer is don't have that kind of noise in there.
Hi. I was wondering how multiband imaging can affect slice timing acquisition.
Yeah. So I'm not the right person to answer that. He is, but this leads me to a slightly different point. Multi-echo images will have a great, I meant to add a slide about this, but then I was sort of going like this and trying to get a baby to sleep. Now, multi-echo is extremely interesting because imagine you do this. If you take, and there are a couple papers out already, and there's one that is going to be out shortly. Imagine you take 3 different echoes at 3 different, shortly, who knows, 15, 25, 40, something like that, milliseconds from the initial injection of the radio frequency pulse. Now, everything that is a typo, everything that is a real T2 star should have a certain decay. Right? You know exactly what is the decay, that things that are induced by the radio frequency pulse and the proton coming back up and pulling out a signal, essentially the Bold effect. You know exactly how that should work. So you can look over these three echoes, and if things don't have a certain shape, then you can say that's probably noise. Right? If something doesn't, for example, sort of come down nicely, just to put it in non-mathematical terms, then it probably is not biological in origin, but it's an artifact. For example, if something had an intensity that wouldn't change over the 3 echoes, then it's probably noise. Right? It's probably non-biological, i.e. noise. So that's a nice way. That's something that I really hope. That seems exciting to me. And Professor Cohen is going to tell you everything about how the multiband is going to help that.
It's a 2-part answer. The first part, sort of initial, we will have a talk on multiband next week. But essentially the idea with multiband sequence is you excite 2 or more slices at the same time. And so if you do, say, a 12-slice acquisition and multiband 4, then you're actually doing 3 sets of 4 slices. Those 3 sets of 4 slices are going to be offset in time, interleaved the way the other ones are here. So you could calculate at what time every single one of these was acquired. But as far as slice timing correction goes, the default slice timing correction is actually agnostic to how the images were actually acquired. It doesn't know the slice timing. So it just interpolates across the time points. And as far as that goes, there'd be no difference in the errors from multiband versus any other sequence.
I'll just add that in my read of the multiband literature with respect to slice
So most multiband studies tend to use very short TRs. That's the point of going multiband, that you can acquire whole brain data in less than 2 seconds, and often on the order of 1 second or less. So most studies, including everything that the Human Connectome Project has been doing, do not do any slice time correction, because when your slices, if you're acquiring, let's say, 4 slices simultaneously or 8 slices simultaneously, and they're only separated by, I don't know, 200 to 500 milliseconds apart, the slice timing correction is not going to help much, but it could hurt by moving noise characteristics around or smearing across slices.
So speaking of the slice time correction, so I'm wondering, so one, instead of correcting it, try to do it, just leave it alone and adjust it during the modeling part? Then
Yeah, that would be sort of, that would be, you know, you would create a slice sensitive model that would be different in its timing for each slice. It's just people haven't done it, but absolutely.
So I have a question about adding motion regressors, like individual time points.
Like individual spikes.
Yeah. So if you are changing degree of freedoms on subject level,
Yep.
How do you really then compare to groups where one group has clearly more versus less? Or how do you pretty much solve it at a group level, in a way?
Yeah, so the answer, the empirical answer is no different. Right? People do not usually get in your vanilla experiment, people do not get concerned with degrees of freedom because you're usually not doing any statistical, you're only estimating at the first level, you're not doing any inference on those maps usually. Unless you're doing a single subject, maybe you're doing a patient report. And then let's say you had 2 patients and you were just sort of doing kind of a comparison. If one of the 2 patients had a lot more, many more spikes than the other one, A, you would know that the 2 patients differ in motion, first of all. But B, if you're then doing significance maps, then yes, one of the two you had fewer degrees of freedom to play with than the other, in which case it would certainly be a problem. People usually don't get concerned with it, A, because you have so many degrees of freedom. You usually have number of volumes -1 to begin with, and you know, at the current sort of at a 2TR, that's about 300 volumes. So even if some 30 of those, you know, go into my regressor as well, so be it. But second, as I said, people don't typically do any inference at that level. So the only degrees of freedom that are relevant when you're doing your vanilla experiment are n, big N. I mean, small n is also relevant, and also tomorrow we're going to get into the details of how small n also plays an important part. But people usually are just not concerned with it in that way, you know, at the first level.
I was just kind of wondering why correction for physiological noise and sources isn't considered part of the standard pre-processing, especially given kind of you can have secondary effects with change in heart rate, change in respiration, things like that.
Yeah, you're right. I mean, it's so easy to acquire. You just put a little pulse ox here on a foot and then write a little stripe here to capture. So all I can tell you is my introspection on it. Having seen some data where it made very little difference, I've never seen it make a huge difference. Not to say that it couldn't in some cases, but when it has been acquired, for example, when I've acquired it, when it was elsewhere, I've never seen much of a difference made. Now the question is, if you did see much of a difference, then is that data good to begin with? Right, so then you get into these kind of hairy decisions. I just don't see people use it often, and when they have used it, I just haven't seen that much. It should make a big difference, but I just haven't seen it. I'm sure it could, but yeah.
Logical noise. Yep. So I assume, you know, inside the brain there's a time delay, different part, you know, according to how far from the blood vessels or this. So
where in the brain also?
Yeah, So then how to take these things into account?
Look, you think that you could model a lot of it, but I absolutely agree. In fact, that could be the reason why it doesn't make as much of a difference, because it's a bit of a cruder measure with respect to the complexities of how this translates, or maybe how the heartbeat rather translates. Yeah, good point.
I was wondering how the motion correction procedures that you talked about might do better or worse with spikes in the motion versus like a low frequency kind of drift.
Wait, I'm sorry. Versus
like someone moving slowly throughout the scan away from where they were initially. Oh, you mean if you have like a spiky versus a slow? Exactly.
I'm sorry? Yeah, right. Somebody sort of like going versus somebody slowly creeping. So if you've ever used those NASA pillows that at my first institution we loved and we thought those were the best for subjects, you get a slow sink, right? Do you know the foamy pillows where it's slowly? So since we thought of that, they've discouraged the use of them. Here, the big jerks will produce big problems, and in particular, they will keep giving you very big spin history effects. The slow drift is also going to give you that, but because it's slow, sort of at each tiny sub-millimeter of motion, it's going to be less problematic. I don't think that you should be any less concerned if you had if I had a gun to my head and were asked to choose, I'd definitely get the slow drift between the two. But of course, they're both bad. Now, don't put the gun to my head. I'll pick one, regardless.
I had sort of a related question, just thinking about integrating the different motion correction strategies that you talked about, and if we're applying this sort of spike regressors and controlling for motion, and can you salvage subjects that maybe show a large degree of motion, or where, how do you make the determination that this is just so much that they have to be thrown out? And does that differ between task-based versus task-free analysis?
So, no motion, keep. Yes motion, bad. Now, how bad is bad? I can tell you that if you read the literature, most people will say, well, anything that has an average displacement of less than 3 millimeters, I'm going to keep. Now, if you're doing a block design where you're hammering visual cortex with a flickering checkerboard, sure, motion will do something, but you're less concerned that if you're having maybe either a quick event-related design or a task-free design. If you have a task-free design, if you keep it slightly backwards, it won't point to that, and it will not get feedback. The task-free design and the ensuing analysis that people typically do after that have been shown to be so damn sensitive to motion that you probably want to be more careful. Not to say that you shouldn't be just as careful in a task-based design, but the effects of it are so disruptive. I mean, your result will change completely. In a task-based, there will be differences. I don't know if it will wipe it out in the same way. In fact, I don't think. I haven't seen it wipe it out in the same way. One thing that you can do, you can just chop. Imagine you have 300 volumes acquired, and there was a big jump at volume 282. Then you can just chop it off and sort of call it, the first 281 your data. Of course, then that is going to introduce some difference, because maybe if you're doing a correlational type analysis, the correlations will be different. But overall, you do know that correlations, after about 5 minutes of data, your correlations tend to be vaguely stable, at least at the level of big default, big networks. Whether that is the same true also in graph theory type analysis, not sure. So it's all of this is a judgment call, except your reviewer will have something to say about it for sure.
Let's do only one more, and leave enough time for lunch and getting to the next talk.
Hi. I have seen the intensity normalization in the order, but
yes.
I think you do not mention that. I have one question for that. That is, when you do the intensity normalization, can you take account of the intensity difference between the cortical region and the subcortical region?
So there are two types of intensity normalization, and I'm not sure which of the two you're thinking of. One is what people do, which is also referred to as grand mean scaling. You take the whole time series and you force the average of the whole time series to be at a certain number. For example, if a cell forces the average across the whole brain or whole time series to be at, I think, 100 square. The other one...
And that is all the voxels?
Yes, the average of everything. The
Okay,
full
okay.
time series across all voxels, yes. It does not take into account cortex versus non-cortex versus subcortical versus intensities at white matter. If you're thinking instead of the intensity normalization where you force every single volume to have the same average across the whole head, then that is a no-no. That sort of is vestigial from PET, where the intensity of the tracer was decaying over time, so people would adjust for it that way. But for example, if a cell used to have this second type of intensity normalization, they since removed from the GUI the button to do it. And you shouldn't do it. There are quite a few published papers showing that it gives big differences, and it's not a good way of fixing any artifact.
Okay, great.
Lunch it is.
Announcements from here. So our after lunch lecture takes place in a different room in the Neuroscience Research Building. And we have to leave here by about 20 after in order to make sure that we're there in plenty of time for Dr. Poldrak's talk. So for those of you who know to get to the NRB, feel free to take other students here who know and drag them over. And other people will leave from this room at 20 after. And I have one other quickie announcement for those of you who are not in the room and are watching from home. We now have a live chat link available if you want to try and send questions. And you'll get there by clicking on the CTRL link on our schedule page, which opens up the videos that we're streaming live. And then if you click on the YouTube button that's at the bottom of those videos, you'll be taken to a place where you can ask questions over chat. Okay, see you all in a minute. Thanks, Martin.